So, good morning, everyone. Respected distinguished speakers, Honorable Founder President Dr. Ashok Kumar Chauhan, respected Chancellor of Amity University, Uttar Pradesh, Dr. Atul Chauhan, sir, respected Vice Chancellor, Ma'am Dr. Balvinder Shukla, Madam, okay, so I should start with Dr. Laurie. उसके बाद Dr. Laurie introduction बता सकते हैं. So, we'll first start the session with the talk of Dr. Laurie, and he will address us on cancer stem cell metabolism. So, I'll request Dr. Vineet to coordinate the session. So, good morning, Laurie. This is Dr. Vineet, and thank you for joining. Okay. Doctor, hello, Dr. Laurie. How are you? So I thought maybe it is. I thought it is late. So, but anyway, we're thankful that you could make it. So, Dr. Laurie is professor in Broad Institute of Cancer Stem Cells, and he's also a director for Technology Transfer Division in UCLA. So, Professor Laurie mainly works on stem cells and follicular stem cells, and he has published several papers in high impact journals in like Nature Cell Science on different aspects of uh, stem cells. So now I would request Professor Laurie to deliver his talk.
Great. I will share my slide. I had to request to share slides. Can someone accept it?
All the way.
All right. Can you see my slides, everybody?
Yes, sir.
Great. Thank you. Well, it's a pleasure to be virtually back at Amity University. It's always a treat to talk about our work and to interact with the folks over there. And so today I'm going to tell you about a story that expands on something I first described there about a year and a half ago when I was there for the EMBO meeting on cancer stem cells. And I presented some data back then to say that cancer cells, as derived from hair follicle stem cells, show a certain degree of metabolic flexibility. And since that time, we followed up and uncovered some mechanisms of flexibility. And so I'll jump right in. So the thing my lab is interested in is this basic question of where do cancers come from? So when you look at a pathology slide of a cancer, you can see normal tissue is off to the left there, very thin but stratified epithelium in the skin. And then off to the right, you can see that that tissue has dramatically expanded. You have changes in cell fate and differentiation and migration. The epithelial cells have now invaded into the underlying mesenchyme. And so a lot of work has gone into looking at the pathology of these tumors, but the pathology is really a retrospective look at what has already happened. It doesn't tell you much about how the tumor got there. And so I'm always thinking about if you could look at this piece of tissue several years before this process formed. And so my lab has taken a prospective approach using molecular genetics to deliver oncogenic stimuli to different types of cell within the epidermis to see which kind of cell can give rise to such structures as you see here. And so this led to a series of studies where we use different promoters to drive the expression of Cre recombinase, to use recombination to either perform a gain of function or a loss of function manipulation in the hair follicle. And we have a variety of promoters. Here are some listed. We've used this LGR6, which marks cells up here on the upper follicle. We've used keratin 15, which labels the bottom of the follicle, and LGR5, which only labels the very bottom of the follicle. And we've used those promoters to drive expression of constitutively active RAS or to delete P53. And then we simply initiate the recombination and wait for tumors to form. And then we can capture tumors along the way, whereby we can, because it's inducible, we can capture the cells and perform molecular or metabolic analyses along the way, namely days after, weeks after, months after. And so we can get an idea of what are the early versus late occurrences in tumor formation. And then we can ask, using the system, what happens if you drive tumors with KRAS versus what happens if you drive tumors with HRAS? What happens if you drive tumors from the top of the follicle versus the bottom of the follicle, etc. And so because of this, we've been able to identify that the cancer cells of origin for squamous cell carcinoma are really this population, the blue population in the middle there, which are the most quiescent stem cells in the follicle. And so this is somewhat interesting that the most quiescent cells in the tissue are actually the only ones that appear to be able to initiate a tumor, which is of course filled with very quickly dividing cells. And so again, it gets back to that idea that if you simply only look at the end stage, namely the existing tumor, it doesn't tell you much about where it came from, because if it did, you might expect that the most rapidly dividing cells of the tissue would be the ones giving rise to the tumor. But in fact, we've found just the opposite. And so another thing I talked about last time is that the hair follicle stem cells sit in a niche that's called the bulge here. This is a resting follicle. A follicle like this will stay like this for weeks in a healthy individual, months or years in someone with a condition like androgenetic alopecia or hair loss, male pattern baldness. And the stem cells are here. And we discovered a while back, when trying to identify metabolic changes that were happening in cancer, we ended up finding that in fact there's an interesting metabolic pattern in hair follicle stem cells just in the normal state. So this is an activity assay that comes out with a purple stain, and so the darkest purple cells are the ones with the most LDH activity. And so you see the most activity here in the bulge where the stem cells are, and down here is a couple layers of muscle tissue, which are known to be highly glycolytic. And then if you sort cells, if you sort these cells out of the follicle and perform metabolomics, what you find is generally a higher level of glycolytic metabolites in the hair follicle stem cells as opposed to the total epidermis. But you don't see much of a difference in the TCA cycle metabolites. So it appears as though the hair follicle stem cells have a slightly elevated level of glycolysis, which might remind you of what happens in cancers, where cancers are known to, of course, have a very high level of lactate production due to elevated increased glucose uptake. So first defined by Otto von Warburg about 100 years ago, he showed that tumor cells or tumor tissue takes up a lot of glucose and uses it to make a lot of lactate, whereas normal tissue takes up glucose to make pyruvate, which is then shuttled into the mitochondria to power the well-known TCA cycle, which of course is responsible for making a lot of ATP. And so my lab was quite interested in this pattern by which the hair follicle stem cells had a very high activity of this LDH enzyme. And so we spent some time trying to characterize it both in the homeostatic state for a hair follicle stem cell, but also in cancer cells. And as just mentioned, here's a little bit of a description that differentiated tissue does a lot of glucose metabolism to power the TCA cycle, to make lots of ATP. It does very little glycolysis, but proliferative tissue or tumor tissue actually does a lot of glycolysis to make lactate, despite the fact that that process only makes a little bit of ATP compared to normal tissue where the TCA cycle is making a ton of ATP. And so you might also know of cancer metabolism from experiments like this, where people use this molecule FDG, and specifically a fluoridated FDG molecule, which is a glucose analog. And if you treat a patient with this and undergo PET imaging, what you find is that the FDG accumulates in tumors, and you can use this to track tumors either at the initial site or distant metastases you see in these little spots for melanoma. And so we know that cancers take up a lot of glucose and use it to make lactate. We didn't really know why, but a few years ago we set out to understand this a little bit better using, again, the hair follicle stem cell as a model. And what you see here is, again, hair follicle stem cells in the normal setting here in this little niche, highly active for LDH activity. You see all the purple stain. And then when you use those stem cells to initiate a tumor and create a low-grade tumor and do that LDH activity assay, you see a ton of activity there as well. Even the low-grade tumor has a lot of LDH activity. Same goes for a medium-grade or a high-grade tumor. And so it made us begin to wonder, well, is the tumor, the high-grade, really glycolytic, or does it have high LDH activity simply because the cell that started it did? And so understand the role of LDH and lactate production in the ability of these cells to make a tumor. And so what we did was created, again, this transgenic model where we have different promoters, in this case a stem cell specific promoter, driving expression of an inducible Cree, and use this coupled with an oncogenic, constitutively active KRAS allele, and coupled that to deletion of P53. And so in a normal hair follicle stem cell that expresses LDH, you have tumor formation, it forms just fine. And then here in this experiment, we did the same thing, but at the same time, we deleted LDH. And so we blocked the ability of the cells to convert pyruvate to lactate, to basically ask what happens, what's the dependence of the stem cell on lactate production in order to make a tumor. And in fact, the tumors formed just fine. We saw the same number of tumors. There's no change in the timing, the degree, the pathology, the immune response, everything was identical. We did see an expansion of hair follicle stem cell markers consistent with this idea that perhaps the tumor has high LDH activity because the cell from whence it came had high activity. And so this is sort of where I left this group last time in describing that there must be some metabolic flexibility to these cells because these stem cells here had no ability to convert pyruvate to lactate. And as a result, they actually stopped taking up glucose. I'm not going to spend too much time on it here, but what we found was when you delete this LDH enzyme, not only did you stop converting pyruvate to lactate, but all the steps above in this glucose utilization cycle also became blocked, as though the cell knew that it wasn't able to convert pyruvate to lactate. And so it thought, well, I'll no longer bother taking up glucose because I'm not going to turn that glucose eventually into lactate. And so this was a striking metabolic response to the loss of LDH activity, and it made us realize that one, tumors are flexible, but two, maybe glucose isn't absolutely necessary for tumorigenesis. And so we started taking advantage of metabolic tracing, where we take either C13 labeled glutamine, C13 labeled glucose, or C13 labeled lactate, and we pulse these into the animal and right before harvesting the tumor. And so these tumors get labeled with these metabolites in the live in vivo intact setting. But then when we harvest the tumor, we can extract the metabolites and perform mass spec to try to understand where did the glucose go, where did the lactate go, where did the glutamine go, so we can get an idea of how metabolites are moving through metabolism and looking at flux. And so in our first paper, we showed that, in fact, if you look at labeled glutamine in these tumors, what you find is that the regular tumors that express LDHA do take up some glutamine, and they use it to make some alanine and things downstream in this process called glutaminolysis. But that the LDH null tumors actually took up a lot more glutamine and used it to make much more of those downstream products, suggesting that glutaminolysis, or the utilization of glutamine, was really boosted when you blocked glucose utilization. And so it looks like this, where glutamine can come into the cell through a transporter, where it's then converted to glutamate by this enzyme GLS, and glutamate can then be converted to alpha-ketoglutarate, which can then shuttle right into the TCA cycle to power the TCA cycle. So even if you have no glucose uptake, you can still power the TCA cycle if you're able to take up glutamine. And it appears as though this is what was happening in the LDH null tumors. And so we took a step back and asked, well, what's happening with glutaminolysis in cancers just by itself? And it had been described, namely the enzyme GLS that converts glutamine to glutamate, has been described to be upregulated in basically every cancer, much like the Warburg effect has been shown to be upregulated in every cancer. And we performed some metabolomics to compare papilloma to skin and found again the products of glutaminolysis were highly induced in squamous cell carcinoma. And so we set out to delete glutaminase. And again, this is just by itself, blocking glutaminase in tumors. And so we took the same approach with the inducible Cre recombinase to delete the GLS enzyme. And in fact, this time we coupled it with chemical carcinogenesis to speed things up. And essentially this consists of adding a mutagen to the backskin to induce mutations, and then following it up with a small molecule that induces proliferation, and eventually you get progression to both papilloma, benign tumors, and squamous cell carcinoma. And so very similar to what we saw with the LDH deletion, deleting GLS by itself, even though this process was upregulated and active in squamous cell carcinoma initiated by hair follicle stem cell, deleting GLS, blocking glutaminolysis, did not affect the time to tumor formation, the number of SCCs, the volume of SCCs. The cancers looked pretty much identical, in the same way that deletion of LDH ended up with identical tumors. And so we've gone a little bit more into the mechanism here, and I'll get to sort of how we think this might be happening. But this was just to show you that we did indeed knock out GLS enzyme. Here it is in the wild type cells. You can see it basically in every cell of the tumor, but we knocked it out, and now these are tumors in vivo. They don't have the GLS. We did activity assays, we did metabolomics, etc. Here's the metabolomics where we use C13 labeled glutamine in those animals, and what you can see is in the absence of GLS, the papilloma or the SCCs that you create indeed are blocked because we can't really detect much glutamine being converted into glutamate or down to asparagine. And so the knockout was indeed effective, but didn't lead to a phenotype in terms of the tumors. And so we started looking at more metabolites here. And what we looked at here was if you pulse the animals instead with labeled lactate, C13 labeled lactate, what you find is immediately the GLS knockout tumors actually take up more lactate. And apparently they use that lactate to power this gluconeogenesis, to go backwards essentially through LDH, to then pyruvate, to make new glucose. And so all these products end up at a higher level. And this has been known, in fact, Ralph Dipardenitis had published that human tumors actually, in some cancers, take up more lactate than glucose to power the TCA cycle. And so that same process we found happening here, but it was induced in the absence of GLS, can be transferred either in or out of the cell through two different transporters, the MCT1 or MCT4. And so in the top panels, you see MCT1. It's expressed in tumors, both in wild type and knockout, the same. We didn't see any consistent effect. But we found actually a strong induction of MCT4 expression at the membrane in tumors. And so that's also just quantified here in terms of glutamine uptake. I showed you this before. This is a new set of experiments done more recently in the LDH knockout tumors that glutamine uptake, and this is again with C13 labeled glutamine, is increased, and therefore the production of glutamate is increased. So just the converse of the GLS knockout experiment, where more lactate uptake was observed. And so in these LDH deletion tumors, going back to those that I started with, where glutamine was taken up at a higher level and utilized at a higher level, we looked at the transporter for glutamine, namely SLC185, and we found actually a really strong increase in expression of that transporter at the membrane of specifically the LDH null tumors. And so similar to when glutamylysis was blocked out MPC1, what we found is pyruvate can accumulate and you end up generating more lactate through the action of LDH. And so we went back to MPC knockout tumors, where again, blocking MPC, we thought that was going to have an effect on tumorigenesis. We previously published that it did not. There was no effect on the rate or degree of tumorigenesis. But what we found was when you knock out MPC, you have an increase in glucose uptake, okay? And an increase in lactate uptake, and an increase in glutamine and glutamate production. And so this MPC deletion, again, didn't have an effect on tumorigenesis, but it did have an effect on the metabolism, namely making the cell think that it needs to take up more nutrients, and so probably upregulating those transporters. And so now I've mentioned the transporters a fair bit, and we went looking to see how do we get a change in the regulation of glutamine, glucose, or lactate transporters. And the striking thing we found, to my surprise, was the obvious experiment was to do RNA-seq on each of these tumors and ask, okay, how are these tumors upregulating the transporters in response to these metabolic manipulations? And in fact, we saw no gene expression change on the transporters. None of them were changed at all at the RNA level. So it turns out all those different transporter regulations that I showed you are actually post-transcriptionally, probably post-translationally regulated. So there was no difference at the RNA level. So I was quite surprised by that. And so we started looking at this a little deeper, and now back to the MPC and the LDH knockout, where I told you that in the MPC, you had more glucose uptake. And in fact, when you look at the glucose transporter, GLUT1, it's strongly upregulated in those MPC knockout tumors. And then in LDH knockout tumors, where I told you that they stop taking up glucose, you have almost a complete loss of GLUT1 expression. And again, all this is at the post-translational level. There's no change in the RNA, but it appears to be super dynamic, and the cells seem to know which nutrients they have access to or not, and are adjusting accordingly, specifically at the transporter level. And so the GLUT1 transporter has been described in the literature a fair bit, and in terms of how does it get to the membrane. And it's known to be shuttled to the membrane by complex regulation on vesicles that shuttle the glucose transporter to the cell membrane or not. And that's known to be regulated by a protein called TXNP. And so what we started doing was going back and looking at these tumors that were made in different genetic backgrounds and just looking at the GLUT1 expression. And so you see the stain here by IHC. It's a very strong pattern, but it's also a mixed pattern. Parts of the tumor have a very high concentration of GLUT1 expression, and parts of it just adjacent have almost no GLUT1 expression. So the interesting thing we found was a complete anti-correlation with the TXNP. And so this is the same area and serial sections of the same tumor, and you can see everywhere you have high GLUT1, you have low TXNP, and conversely, everywhere you have low GLUT1, you have high TXNP. They're completely anti-correlated. And so it appears as though, as others had previously described, that TXNP is probably regulating the membrane presentation of the glucose transporter. But then you look in the LDH knockout, and I told you that in the LDH knockout, they stop taking up glucose and they have diminished glucose transporter. And you see that here in the tumor. You see generally there's patches that have some, but generally it's much lower than in the wild type. And you see, conversely, strong upregulation of TXNP expression throughout the tumor. And so again, you still have the anti-correlation, and because you have more TXNP in the LDH knockout, that's presumably why you have diminished glucose transporter in the knockout, and that's just quantified off over here to the right. Similarly, I told you that the MPC knockout has an increased glucose transporter expression at the membrane, an increased glucose uptake, and you see that as well with this anti-correlation, where in the MPC knockout you have diminished TXNP and increased glucose transporter. So it's the complete opposite pattern you can see in the quantification versus the LDH knockout. And so we've been thinking a lot about how is this happening? How are these transporters getting to the membrane and being kept there? How does the cell know that it needs to put them there in order to take up a new nutrient because some other nutrient has been starved? And so we've just been quantifying this in every setting. Here again is the glutamine transporter, which basically disappears in the GLS knockout. You can see it there, but it's induced in the LDH knockout, where, remember, they take up more lactate. And this glutamine transporter, there's not a lot of literature like there is for the glucose transporter in terms of how it's regulated at the cell membrane, but there is some data to suggest there's an interaction, a physical interaction with the EGFR receptor, the epidermal growth factor receptor, which is known to be really prominent in, particularly in epithelial cancers. And so what we did here is to look at phospho-EGFR, so namely the active EGFR, after it's engaged ligand, the receptor gets phosphorylated, autophosphorylated, and becomes active and starts acting on targets. And so what we found was this interesting subcellular localization of the EGFR. In the absence of the GLS, where the receptor for the glutamine transporter is lost, you have this different intracellular distribution of the EGFR, namely you lose it at the membrane. So quantified over here, if you can't see in the image, the EGFR at the membrane is diminished, and that correlates with loss of the glutamine receptor at the membrane. Vice versa, in the LDH knockout, where we had an increase in the glutamine uptake and glutamine transporter at the membrane, you have increased phospho EGFR signal quantified over here. And so while it's not as definitive as the GLUT1 TIC-SNP interaction, it starts to get at this idea that the cell has evolved ways to physically put transporters at the membrane in times of need. And so all those data are summarized here, where essentially in the LDH knockout, you stop taking up glucose, and there's interaction of TIC-SNP and glutamine determines how much glucose transporters at the membrane. But in the absence of this glucose uptake, you take up more glutamine. We think that the transporter is physically brought to the membrane by active EGFR signaling, which is interesting in a variety of settings. A very prominent cancer drug, erlotinib, targets the EGFR receptor activity. And so it's interesting to think about what's happening. And we haven't tried those drugs in this setting, but it's interesting to see what happens with EGFR inhibition, whether that regulates the glutamine transporter expression at the membrane. And then I mentioned over here in the GLS knockout, you start taking up more lactate through the MCT4 transporter. In this case, we have actually no idea how the MCT4 transporters is trafficked to the membrane. We're actively investigating. But I'm just fascinated by how the cell appears to know that it's lost one of these nutrients, and instead of turning on some transcriptional program to try to make more RNA and protein for the transporter, it simply physically puts more of that transporter at the membrane, basically opening a door to the extracellular environment where there's high concentrations of these nutrients, and it just takes up more. And so to finish, I'll tell you that we went a step further to test the limits of this metabolic flexibility by knocking out both GLS and LDHA in the same mice, then following up with the DMBA TPA protocol. And the results were pretty striking in that there were papillomas that could form in the double knockout. But by the end of the experiment, they were all gone. So we couldn't count any at the end. And we never got any SCC. So this is unlike the GLS knockout or the LDH knockout, which reliably still made papillomas, reliably still made NPCs, or SCCs, excuse me. In the double, we would get some papillomas that either regress or they die off or fall off. It was a quite striking result. So there is a limit to this metabolic flexibility, and essentially we found a combination that can block tumorigenesis from hair follicle stem cells. And so to finish, I'll tell you about the last set of experiments we did, where we're trying to make the case that the tumor cell, probably the reason that a lot of approaches to treat cancer by blocking a metabolic pathway have failed, is because of this metabolic flexibility. And our data argue that at least some aspect of this metabolic flexibility is due to the ability of the cell to put more of these metabolic nutrient transporters at the membrane. So to test the idea that membrane transport is important for the metabolic flexibility, what we did was take advantage of this MCT4 inhibitor, AZD0095. And we did that coupled to a GLS knockout. So the theory goes that the AZD, a drug which failed in the clinic, by itself, in theory, should not do much to tumorigenesis in this model, unless you've also knocked out the GLS. Because when you knock out the GLS, you take up more lactate to power the TCA cycle, And so what happens if you use this drug to block lactate transport both in the wild type or the knockout setting? And so what we did was we essentially tracked tumors over time. And so we initiated tumors the same way I just described with DMBA, TPA. And the red line is the knockout, and the blue line is the wild type. We gave all the animals the transporter inhibitor to ask how do the different kinds of tumors respond. And so what you can see in the wild type, the blue line, is you start tumorigenesis, and it basically just keeps increasing. Here, this tumor score is meant to indicate sort of tumors are growing, they're converting from papilloma to SCC. So in the wild type setting, the tumors grow, continue to get more aggressive, etc. But in the GLS knockout, at first, the tumors are growing at exactly the same rate as the wild type. But once we gave the drug here at day 137, now the tumor aggression levels off completely, and the tumors don't progress. And this only happens in the GLS knockout, suggesting that in those tumors, they sensed disturbance, essentially, in their glutamine utilization, started taking up more lactate, but then when we gave them the drug, now they couldn't take up lactate, and they weren't metabolically flexible enough to overcome that, and so tumorigenesis sort of flattened out. And you can see over here, some tumors even completely regressed, as judged by the sort of negative slope here. And so this is consistent with the idea that that tumor presentation at the membrane is a key for this metabolic flexibility. And so just to summarize what I've told you, initially we described this idea that hair follicle stem cells are more glycolytic than their progeny, exhibiting evidence of the Warburg effect. I didn't tell you a bunch of work that's gone on to describe how you can take advantage of this to activate stem cells to make hair grow. We've started a company, and we're in clinical trials with MPC inhibitors to do that. But I also showed you that hair follicle stem cells are cells of origin for squamous cell carcinoma, which are also highly glycolytic. And so our initial expectation was that blocking glycolysis in the stem cells would affect their ability to start tumors, but in fact it did not. The tumors don't even need glucose, in fact. But they compensate, it appears, by utilizing glutaminolysis. Separately, we now showed you that tumors are not affected by themselves by loss of glutaminolysis. Again, glutaminolysis had been the subject of companies that had started trying to treat cancer. One company, Calathera, here in California, failed. This single agent was unable to have an effect because we think that glutaminolysis null tumors simply take up lactate as an alternative energy source. And that all this flexibility that I described today, not necessarily all metabolic flexibility, but the flexibility I described today, all appears to be due to changes in surface expression of metabolic transporters. And then all that said, if you block both glycolysis and glutaminolysis, you can prevent tumor formation. And I like to think that this work will someday lead to potentially targeting transporters, but it'll really have to be combinations of transporters in order to effectively treat cancer. And so now, going forward, we're trying various combinations of both enzymatic inhibitors and transporter inhibitors to try to understand if there's potential efficacious ways to treat cancer taking advantage of this. We're looking at these compensatory metabolic pathways. How do the cells know that they've lost a metabolite? How do they post-transcriptionally put those metabolite transporters out of the membrane? And then, what is that sensing mechanism? And so I'd like to acknowledge Carlos Galvan, who's done most of the work I described today. He's defending his thesis in a couple weeks. I'm very proud of him. He's done this work with a variety of undergraduates in the lab. I'm pleased to acknowledge Heather Christoph. Her lab performs a lot of the in situ analyses, metabolomics work with us. I didn't get to talk about chemistry today, but we do that with the Jung lab and in situ metabolism with the Collar lab, and our informatics work is supplemented by a group at USC led by Nick Graham. I didn't get a chance to talk about adrenergic signaling, and then like to acknowledge our funding sources. And I'm happy to take some questions if there's time. I wasn't looking at the clock at all.
Thank you.
Thanks for your attention.
Thank you, Professor Lowry. And it's such a fascinating work. And it shows that how cancer cells are, they're so flexible that if you block one pathway, they can take up another one. So, for example, LDH is blocked, so they rely on glutamine. So, it's very, very fascinating research. So, now it's open for questions. Mike. So, any question from the students or?
This presentation, William, it's very knowledgeable. In our audience, actually, there is several postgraduate and graduate students also there. So, for them, just I want to highlight one thing because it's very, you have nicely presented. That is a cancer cell is taking advantage of the glucose transporter, glutamine transporter, lactase transporter that you are trying to block it by GLS, then LDHA, and you are blocking and you are watching the cancer progression in the mice, knockout mice even. So thing is that there is a when you are lastly talking about the pharmaceutical inhibitor that is you are going to use. So how you can differentiate the inhibitor effect on the cancer or cancer stem cells with the normal cells or primary cells?
Yeah, it's a great question. It's something I worry about as we do these experiments. Typically, what we're doing is we're topically um, uh, formulating the drugs and topically applying them, and lots of cells are exposed to them. And so, what we do is we start by taking normal mice and taking each of those inhibitors separately, treating, and then looking at the activity of the targets of each of those inhibitors. And what we found is that just adding any one of them to the skin, to normal skin, doesn't appear to have a huge effect on homeostasis in the skin. And, some of them are inflammatory, and so those we try to avoid. Um, but what we typically can do by taking advantage of the skin is that we can start by taking animals where the tumors have already started, and so they're visible. And so we can physically just paint the tumors as opposed to the surrounding unaffected tissue. And so, really, we haven't seen much of an effect of these drugs on the normal cells. I do think that, in fact, the tumor cells appear to be more sensitive to some of these metabolic manipulations than normal cells. We don't totally understand why. There's interesting data out now showing from Josh Rabinowitz at Princeton, actually showing that cancer cells actually have slower TCA cycle metabolism. They actually make less ATP. And so my feeling is that this cancer cells might actually be more sensitive to some of these drugs because each of these drugs is going to inhibit ATP production in different ways. But if you inhibit ATP production in a cell that already has diminished ATP, I feel like you probably get it down below the threshold at which it needs to create ATP in order to divide. And whereas a cell that has a fully functional mitochondria and is making, the 36 moles of ATP per mole of glucose, has plenty of spare capacity. But the cancer cells don't actually have the kind of spare capacity that I think we thought they did. We all thought that cancer cells are highly metabolic, but in fact, maybe the opposite is true.
Yeah. Any more questions? Yes. So, Dr. William, it was a great work that you have done. It's a voluminous and certainly this will lead to therapeutic applications subsequently. I just wanted your remarks on, because you showed a canvas of transporters, and which one you feel, whether it's a glucose transporter or it is the lactate uptake, which is very essential to give the energy through the glycolytic pathway. So, which one is the most important one? Then another comment is, can you also have a combination of 2-deoxy-D-glucose, which is an analog of glucose, which will sensitize the stem cell or cancer cell initially, and then you give this transporter inhibitor. Will there be a synergy? Can you look at those 2DG in combination with your transporter blocker?
Yeah, it's a great idea. We haven't done that particular combination. Uh, to your question about which is the most important transport, I think it's absolutely going to depend on the cancer and the sort of lifestyle of that cancer. Each one is going to be in a different environment, having access to different concentrations of various nutrients, and so it's going to depend on what's in the surrounding area of the tumor. And then it's going to depend if you manipulate one pathway, it's then going to have an effect on a separate pathway. And so it all depends on which one you first target in terms of which one becomes the most important. But your second question about using 2DG, I think it's a great idea. It would be the equivalent of what we did with the GLS and the lactate transporter combination. We absolutely should do it. We just have not. So maybe someone there can do it and tell me how it goes.
Can I ask one question? AK Singh this side.
Yeah, please, sir.
Great lecture. The comment. Can you say that if we use 2DG, because 2DG has been used for glioblastoma, what happens when you give it systemically? Then it is not only taken up by the tumor cell, but also by the normal cell. And particularly the brain, then the cell gets starved for the energy, and sometimes it really, we are not able to really achieve the target or targeted sensitization. So that has been tried many times, but could not finally become the molecule or the scheme of choice. This is my submission. One. Number two, if I have understood correctly, you said that the metabolism in the cancer cell is lower. As you said that there is the indication. There is the indication. So if it is so, then and if it comes true, then PET, this FDG imaging, this will become obsolete, futile, because the very concept of the FDG uptake is based on the metabolism, higher metabolism in the cancer cell.
Well, I'll push back a little bit. The FDG imaging is based on the uptake of FDG, which actually doesn't tell you how much ATP is being produced. So when I say that tumors have a lower metabolism, it's a blunt way of saying they produce less ATP. And so a cancer cell that takes up a ton of glucose makes less ATP than a normal cell that doesn't take up as much glucose. But that cancer cell on the FDG PET will light up as much brighter than the normal tissue. Not because it's more metabolically active or because it's making more.
Only for the uptake.
Yeah. That's all it's telling you.
Well, actually, this is Prakash here, Ridesh Prakash. I have a point here. Although we are discussing about the tumor metabolism, and sensitizing the tumor for the cancer-directed therapies, mainly radio and chemo. What is more important here is the share of metabolism between the immune and non-immune cells, on cancer cell. Most of the time, if you compensate the cancer cell for the immune for the metabolism, then you are also tweaking the immune cells, And most of the time, if you deprive the glucose uptake by the immune cells, what you are going to do, you're also impacting the tumor immune metabolism, and that is where the immune cells get polarized, and then in that condition, they mostly, rather than controlling the tumor cell, they rather support the tumor cell. They become, pro-tumorous, and with a clear example is a VGF and TGF beta signature in the highly metabolic tumor. They are resistant also. So the tumor endothelium also contribute in this in the share of the immune metabolism. So that is what I personally feel, that if you can manage the metabolism between the tumor cell and non-tumor cell, then I think you can have more impact on the therapy.
I agree. It's a really important point to make, and to my discredit, I've completely ignored the immune system in this talk and in my lab. But I would tell you a couple little pieces. One is that in the squamous cell carcinoma, we do see immune cells, and the immune cells that show up have an even higher glycolytic rate than the tumor cells. They take up even more glucose. They have a higher LDH activity. The other point I'll make is that you see similarly, I'm not sure if they're the same cells or different cells, immune cells show up that have very high GLS expression, meaning they're performing glutaminolysis. And so I didn't point it out, but in the immunostaining where we knocked out the GLS, we only knock out the GLS from the hair follicle stem cells that start the tumor. We're not knocking it out in the immune cells. So the picture was totally blank except for a few little bright spots. Those were immune cells. We haven't knocked it out of them. And so I've glossed over it, but essentially we only knock out all the pathways I'm talking about genetically. All that is only done in the epithelial cells. We haven't made any manipulation in the immune cells. Separately, all those drug treatments target all the cells, whether they're epithelial or immune cells. And so we've completely ignored the immune component. We absolutely need to pay attention to it, though. I'm certain they're playing a role. It's just when you're looking at the metabolism of the whole tissue, by tissue I mean tumor, 99% of it is due to the epithelial cells. The immune cells don't make up
So,
percentage-wise a giant proportion. But I would love to know if we could manipulate the metabolism of the tumor cells to make them more effectively target the epithelial cancer cells. And that's where we haven't started yet. But I appreciate your point.
Thank you. Thank you.
thank you.
Yeah, we are not taking further questions.
Yeah, so we are running out of time. So thank you very much, Professor Lori. It was short time, and you have
Big clap for him. Yeah.
Thank you. I appreciate it.
so. We are extremely thankful that you have accepted our invitation on such a short notice and delivered such a fantastic talk. Thank you.
Thank you.
Thank you.
Bye.
Next.
So, good morning, everyone. Respected distinguished speakers, Honorable Founder President Dr. Ashok Kumar Chauhan sir, respected Chancellor of Amity University, Uttar Pradesh, Dr. Atul Chauhan sir, respected Vice Chancellor Dr. Balvinder Shukla madam, and respected Dr. Shilvamurthy sir, DG ADSI, President STEP, campus heads, all esteemed guests, chairman, directors, head of the institute, faculty members, and all the participants who have joined us for the inaugural session of the International Symposium entitled Cancer Metabolism and Drug Discovery, Role of AI and ML. So I, Dr. Priyanka Jain, Assistant Professor at Amity Institute of Molecular Medicine and Stem Cell Research at Amity University, Uttar Pradesh, welcomes you one and all to this international symposium, which is being hosted by Amity Institute of Molecular Medicine and Stem Cell Research, Amity University, under the guidance of Professor Gudev C. Das, Chairman, Professor Surajit Vishwas, Director, himself, Professor Simran Tandon, Director Research, himself, and this symposium is being organized by Professor Vineet Kumar and myself as a co-organizer. So I invite all dignitaries for the lighting of lamp and Saraswati Vandana.
Yeah, welcome. Speakers also here. Mayank, आ जाओ. आंसर के पहले ना किसी नाम की चला जाए. हाँ, किसी नाम के पहले उसके बाद किसी का सर उसको कैसे सर. हाँ.
या कुन्देन्दुतुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्कर प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
So I welcome the dignitaries on the table. So I welcome Professor Budev C. Das, Chairman of EMSR, Dr. Surajit Vishwas, Director of EMSR, Professor Simran Tandon, Director Research, EMSR, Dr. Vineet Kumar, the organizers of the symposium, EMSR, and Dr. Sulvamurthy Sir, DG ADCI, President STIF at the desk. So I welcome for the address of Professor Balvinder Shukla, Honorable Vice Chancellor of Amity University, Uttar Pradesh. Good morning, ma'am. I request you,
Yeah.
please join us.
Professor William Laurie and Professor Dr. W. Silvamurthy, Professor B.C. Das, Professor Subrajit, Professor Simran Tandon, all the colleagues from EMSR and Amity University. I, on behalf of Honorable Founder President, Honorable Chancellor, additional Vice Chancellors and Deans, head of his institution, extend very warm welcome to all the distinguished speakers for the International Symposium on the theme Cancer Metabolism and Drug Discovery, Role of AI and ML. Just now, the presentation given by Professor William Laurie was excellent, which has given a lot of details about the recent research about the cancer stem cell metabolism. And I'm sure that today's this symposium is going to throw a light on many areas, especially on the cancer metabolism and drug discovery, the role of AI and ML, and which is a testament to the collective pursuit of many researchers and scientists on innovation and commitment to harnessing the power of technology to combat deadly disease, cancer. As many scientists and researchers have assembled here today, which reminds of the formidable challenge that cancer poses on the humanity. Yet, I feel that we all are filled with hope and optimism, knowing that the convergence of AI and ML and cancer research has the potential to revolutionize our understanding and treatment of this disease. Amity University has always been at the forefront of this endeavor, and I'm delighted to see so many of our faculty and students leading the charge, and these researches and discoveries are crucial to our mission of advancing knowledge and improving the lives. That this symposium will delve into the latest advancements in the various areas, whether it's the materials, whether it's the device fabrication or chip design, and we'll explore the cutting edge research, innovative applications, and the future directions in this field. As we are aligned with the sectoral vision of Viksit Bharat at 2047, one side we have the mission of India being the hub for Indian traditional medicine system, the Ayush. At the same time, understanding and recognize the critical role that science, technology, and innovation plays in driving India's growth and development. India to be among the top 5 innovation nations, India to be the hub for the startups, as well as India to be the focusing on the various technologies such as biotechnology, cancer research, space science, quantum computing, semiconductor research, and other areas. So I'm sure this international symposium is going to bring out applications of emerging technologies and our commitment to the vision of the India being the Viksit Bharat. During the day, all the researchers from across the globe will delve into the latest advances in cancer metabolism and drug discovery, exploring the vast potential of AI and ML to transform our approaches. And the experts chosen by the organizing team, Professor Vinit Kumar, and the others, is really very important. And Honorable Founder President's vision is to be, India to be superpower through research, innovation, and beyond. And therefore, Honorable Founder President has always focused how research be not only the responsibility of faculty, but also be the passion of PhD scholars, passion of students, and whether they are the undergraduate students or the postgraduate student, the research has to be integral part of their curriculum. So therefore, right from the graduation, the students start thinking of innovation, thinking of creativity, and which will lead to bringing the innovative, one side the technologies, the practices, but also finding the solution to various important problems which are either addressed or focusing on the societal needs or focusing on the national priorities and making the life better. So once again, I would like to congratulate entire team of EMSR under the dynamic leadership of Dr. B.C. Das, and I'm sure with Dr. Simran Tandon returning back to EMSR will further strengthen Dr. Das' team along with Dr. Biswas and Manoj Garg and many of the leading scientists of this institution and fulfill the dream of Honorable Chancellor that India be the superpower by 2030 and now be the Viksit Bharat by 2047. And I see Dr. Silva Murthy ji is sitting there, who is leading the research for the whole group. So, Dr. Silva Murthy ji, I'm very happy that you could spend time physically in this EMSR and this symposium. I could not come. But I know that the mission which has been given to us by our Honorable Founder President under your leadership at the group level and under the leadership of Dr. Das at the university level, we are going to achieve those goals and really contribute to the Viksit Bharat by 2047. I extend very warm welcome to all the researchers and scientists who have come from across the globe, physically or online. I really thank you and look forward working with you through my researcher and scientists, and we make achieve the goal eradicating the cancer, and that's it needed for the humanity the world over and make this world a better place to live. So thank you very much and all the best to the success of this international symposium.
Thank you, madam. Please.
Thanks a lot, ma'am. So I welcome Professor V.C. Das for his remarkable, valuable comments.
First of all, thank you very much, madam, for addressing this gatherings for this today's half-a-day symposium. Most respected Honorable Founder President Dr. Ashok K. Chauhan, the dynamic chancellor, our Dr. Atul Chauhan, and Madam Vice Chancellor, Professor Dr. Balvinder Shukla, just to listen to her. So, and most importantly, all the speakers, the first speakers from US, Professor William Lowry has started the tone of this meeting. Excellent deliberations and excellent aspect of the research, which just doesn't give information or the study, but it gives the lot of questions. He read as a lot of questions. I know there are 7 questions, but then there are many more questions. Many people will be like to ask, but it is not done because that is the type of study which generally just doesn't answer anything, but it opens a lot of questions. That's a great research. So he has just started the tune of this meeting at the half-day meeting, but I am sure the other speakers who have been selected very carefully, I welcome them all. Professor Rahul Kumar from Department of Biotechnology, IIT Hyderabad. Then I think he's here all the way. Welcome. Thank you so much. And also he has always come many a times, Dr. Chandra Prasad Prakash, Medical Oncology and AIMS, New Delhi. And Dr. Rasmarani was a colleague of us here, but she is at the moment head drug discovery unit and Jubilant Biosys. And then Professor Saran Kumar, Kusuma School of Biological Sciences, IIT Delhi. Everybody knows that is one of the top school in the country. And Mayank Singh, Dr. Mayank Singh, medical oncologist, AIMS, New Delhi. He will be online. Oh, yeah, you are here. So I am confused with the Hyderabad. Okay, Hyderabad is sitting somewhere else. Behind. Okay, thank you. So thank you so much. I know many of these are medical oncologists and AIMS. They are young faculty. I am not known to introduce to know, but I was a regular visitor earlier because whole life I have spent in ICMR and AIMS was almost. So I knew everybody, but they are all old people. They have retired, many of them, and now the new generation has come in. So welcome all the people and the very selected speakers, and their topics are very carefully chosen. I hope it will be a great opportunity for all the students of our graduate, undergraduate, and postgraduate, and PhD students. And you need not to have afraid of. You can ask any nonsense question. Doesn't matter. You are students. You can ask anything, and it is all perfectly fine. And of course, our own colleague and one Professor Selvamurthy, who is the president ASTIF, and all in all for the research and innovations in the Amity University, which is the junoon of our honorable founder president. He is old enough, but then in mind he is young enough to guide us anytime in the day or the night till 12 o'clock or 2 o'clock even. So he comes out with some decision, some idea of the research and innovations, always thinking something, how to do good research. And it is the actual word of the Hindi is the junoon. So he has a junoon. And that is why it is all we are like his soldiers. Whatever order comes, we are ready to do what is impossible and all. But as far as and all my colleagues, Dr. Smriti Biswas, the director, and the new colleagues, Professor Simran Chandan, who was earlier a colleague in the initiation, but then he left to Mohali MIT, but then she is now come back here, and so she is director research. So we will have more strong, and other all professors are here, and all my colleagues in the audience and in the and organizer is Professor Beneath. He did the EMO workshop. You will be knowing on this topic. That's why it is another extension of that topic, the metabolism and the cancer, the drug metabolism. So this International Symposium, Cancer Metabolism and Drug Discovery, role of AI, artificial intelligence, and the machine learning. There are many other topics in that, but the mainly two aspects has been made it that they are very much relevant, and it is made important that the always in amity is in the forefront of research, either in the academics or in the research. So how far we are successful, that depends on that. We try our best to start with the things. So we have a huge facility of the 10 petaflop, the computer facility, and it is several crores have been spent to create that facility. So that's not for the cancer, but major application on cancer and other diseases, and that is already in place, and it is regularly being updated. And so it is now that things have changed, that you can do wonders if you use the AI and machine learning or AI, so that which can be done in long time, but it can be done very quickly and very specifically. So that is what, but then it is just a new science has come in, and there are a lot of enthusiasm, but one has to be very careful how it can be used specifically to detect cancer, design disease, or design drugs and their delivery. So this is all, and that leads to the discovery of the drugs. So this is the main theme of this workshop. I think this is very timely theme, and I hope, and the topics being chosen very apt topic, and their speakers are from the very chosen institutes, and they are extremely involved with the research. They are not only basic scientists, but there are clinicians which are looking in the use of this exactly with the not on the bench, but they are also bench as well as in the bedside. So this is a unique combinations, I would say. So any type of cancer research, it is important to have the people from both, from the man on the bedside of the patients and the man on the bench. So there is a crosstalk between the two people, and also to the students, so that their feeling, their understanding is conveyed to the students and the researcher, so that it comes to the mainly to the things that it should be applied, or application research that which can be, may not be tomorrow, but day after tomorrow, or a year, or a 2 years, or a 5 years, or 10 years time. We should come out with something which will go to the bedside, to the patients. That is very important, and that is with this aim this orchestra has been organized. I thank all the participants and all my colleagues once again, and once again I welcome all the speakers to make it convenient to come over here physically, and to physical speaking and online speaking has a little bit of difference. So it's good that the students can interact with you when you go for a tea and others, or at lunchtime. So thank you very much once again. Welcome to this meeting.
Good morning, Dr. Das.
Sir, please, Chandra,
Thank you so much.
Good morning, Dr. Das.
Thank you, Dr. Tandon. He is our additional Pro Vice Chancellor. He has joined. He also left, his husband of Simran Tandon. So both left to Mohali Amity, or he is back and joined as a additional Pro Vice Chancellor here. Thank you, Tandon sir. Thank you very much.
Thank you.
Thanks a lot to Das sir. So I request Professor Surajit Vishwas for his address to the symposium.
Thank you, Dr. Priyanka. I will not take too much time. because we are running already late. Just I am very thankful to all the guests today and welcome the speakers on our international symposium on cancer metabolism and drug discovery. And especially, we are bridging the cancer metabolism with the machine learning and artificial intelligence. That is a big topic, and I'm very much thankful to the organizer, Dr. Binit, actually bring this proposal just 1 week before, but it happened very fast. But we are trying to do it successfully. So, first of all, I am very honored and getting always blessings from our honorable founder, sir. And he always silently watch all the things. Maybe in between, anytime he will come and give a comments on that. I am very thankful, Dr. Balbinder Shukla, Honorable Vice Chancellor, ma'am. Thank you, ma'am, for your the inputs with for our symposium. I'm thankful to Shilva Murthy sir and BC Das sir and Dr. Shimran Tandon to be here. So this is the only, don't take it the symposium for the scientists, that is because students are also here. So I'll make some small comments on the student, because here PhD students are there, graduate students are there, and postgraduate students, not only from our institute, even from the our domains also. So please speak up and ask the questions to the speaker. So whenever you are trying to ask some question, you'll feel maybe I don't know. But this is the big story of I don't know, because sometime when you'll ask questions to the teacher, they can also tell you that is I don't know. Sometimes that is our internal always feeling. It's bring us powerless. So this is the one of the inspiration talks from the Harvard University actually. But I want to say that is please ask question, even try to find out something what we don't know, because I'm telling confidently, even in the morning you have listened the Williams talk, and the cancer cell are too much clever, because when the scientist is trying to stop the one glucose transporter pathway, it taking the another transporter, that is lactose, otherwise it's taking another transporter, so it is very difficult to block. Even we don't know exactly what is the molecular mechanism, what is the metabolism going on in the cancer cells. We don't know. Even when the doctors is prescribing some of the drugs, and metabolism inside us, even it varies to not only age to age. Even I can give you one small example, like metabolic one athletics, maybe it is in 50 years one athletics. His metabolism is comparison with the maybe 150-year boy or girls. So very fast metabolism in there. But in terms of the age, cells behaving differently. So treatment also very personalized. So doctor also don't anything. So no. So please come up with enjoy the symposium session and ask the question and get each and every the aspect of the research over here will be discussed by our the speakers. Thank you very much. Thank you. Oh yeah. Sir, Dr. Chandradip Tandon, sir, welcome and again back in Amity University Uttar Pradesh campus. So it is great if you say a few words regarding our symposium, and because you are always respected for us, and for me especially. So please give some inputs and your comments.
Thank you, Dr. Viswas. Very good morning to you and all my colleagues here. It's an opportunity for me. I mean, it's always an honor to be back in such a prestigious university. And this topic of cancer, am I audible? I'm audible, Dr. Viswas?
Yes, sir. Yes, sir. Yeah. You are
okay.
Yes.
I guess this topic is very important because, these days we see that there is an increase in each, and if you talk of any family or any family member, every family has one or two cases. Unfortunately, people are suffering from cancer, and there are lots of researches also going on, and also have already taken place, in which all pathways have been discovered. Now time has come for a translational part of it. Instead of just resorting only to chemotherapy or radiotherapy, which are currently the modes of treatment of cancer biology for cancer pathology, if AI or ML can provide a way forward, that would be great, so that the drug discovery, which is one of the subtopics here, along with cancer metabolism, can see light of the day. So we all look forward to this, and I'm very sure the deliberations which will take place amongst all the eminent scientists and the students is going to be very fruitful. Thank you so much, and wish you all the best.
Thank you, sir. Thank you, sir. Dr. Priyanka, please continue.
Thank you, Tandon sir and Vishwas sir. So I welcome the address by Professor Simran Tandon, Director Research, sir. Good morning, everyone. I feel very fortunate to have come back at a time when such a wonderful international symposium is taking place. Now, most of you have already heard Dr. Laurie, wherein he said that, and as echoed by Dr. Biswas and by Dr. Vineet, that the cancer cell somehow, whatever challenges are thrown in its path, it can make certain mutations or modulate pathways so that it does not stop its spread, its proliferation, and its metastasis to each and every part of the body. Now, what is basically required is that some kind of tool which is going to accelerate cancer management and thereby help in alleviating or reducing the symptoms and strategies to reverse cancer. Now, keeping this in mind, this symposia, wherein artificial intelligence and machine learning is a very powerful tool, this is going to help accelerate both drug discovery as well as strategies to combat and stop the spread of cancer. And today we're very fortunate that we have such vibrant and young researchers who have ample experience in this field, who are going to throw new light into how their strategies and their research can help in the management of cancer. Thank you so much. Thank you, Simran ma'am. So now I welcome the address of Dr. Silvamurthy sir, DGA DCI and President STEF, for his valuable remarks.
So good morning, students, all the delegates, invitees, guests. Let me first of all understand how many of the students here are from PhD. Just raise your hands. How many are PhD scholars? Very good. How many of you are doing masters in molecular medicine and stem cell research? AIMSR, master students, and the rest are undergraduate. How many undergraduate students are there? Great. So let's clap for all the brilliant students who have attended here. It's by you. The whole seminar which is being conducted, all the experts who have come all the way from All India Institute of Medical Sciences, industry, and every one of us here wants you to be groomed as future leaders. So this seminar is for you. So let me also convey the blessings of Honorable Founder President, Dr. Ashok Chauhanji, and for him, it is the research and innovation is his life. So the whole focus he gives in the whole Amity Education Group, which is today 12 universities in India, 18 campuses abroad, 28 schools. So the research and innovation is the fulcrum, is the pivotal point in which the whole education is revolving around in Amity Group. And so for him, it's the most one of the satisfying moments to see the brilliant students, PhD scholars attending and listening to all the achievers who have contributed to the field of cancer biology and also cancer metabolism, cancer therapeutics, and looking at a new paradigm in terms of applying the artificial intelligence, machine learning, deep learning, all those aspects. So founder president is also listening to the whole program from the beginning, and so he will be very happy to see the deliberations coming to a fruitful outcome because he has set a mission outcome. Because
conferences are held, symposia are held, seminars are held. So what is the ultimate outcome? How does it benefit the cancer treatment? How does it help the cancer patients for early diagnosis or for therapeutic application? So I'm sure this seminar will also throw up some new avenues and also different. And ultimately, that will be the outcome that this is the path in which the country should move ahead, the whole globe should move ahead. So we will be eagerly awaiting the outcome from Subhajit Biswas, who is the architect of this program, along with Vineet. So let's give a big clap to these two stalwarts in all in Amit Institute of Molecular Medicine and Stem Cell Research, under the able guidance of our great leader, Dr. B.C. Das, and who is globally known as expert in the field of cancer biology. He worked in Germany with the Nobel laureate who discovered the HPV, human papilloma virus, which causes the cancer cervix. And so he is driving. He has brought all big people, stalwarts in the group in different cancers, be it breast cancer, cancer cervix, and head and neck cancer, oral cancer, liver cancer, pancreatic cancer, you name everything. The experts are here. And he has also assembled a large number of Ramalinga Swami fellows. So these are the prestigious fellowships and also the Welcome Trust fellows. So thank you. He's like a magnet attracting the best brains to his group. And he has developed a big group. Now Dr. Simran Tandon has come back to his canvas of the experts in the field of molecular medicine and stem cell research. Thank you, Dr. Das, for all that wonderful work that you are doing. And even in the vaccine, which was released recently for the cancer cervix, and Dr. Das' signature and his immense contribution is there. Let's clap for Dr. B.C. Das, whom we are very proud of. And also, I would appreciate the whole team of experts, starting from Subhajit Biswas, Professor Simran Tandon, Vineet Kumar, Manoj Garg, Priyanka, who is conducting the program, and all of you. And I'm not meaning Shinjini is here, and every one of you are contributing immensely to this cancer biology. Now, let me share some thoughts that what is that we would like to look at. I would like to emphasize on three important things. The first is integrative medicine. Because India had the traditional systems like yoga, Ayurveda, Siddha, Unani, and all these were there 5,000 years ago. So now how do we integrate these traditional systems of our own country with the modern medicine? So Dr. Das himself is taking the effort first. I would like to narrate one of the research which is happening. It's a triple conjugate which Dr. Das has developed, which is a known anti-tumor agent which is highly chemotoxic, and curcumin, which tones down the side effects of the known anti-tumor agent, folate, which gives the signature of the cancer stem cell. She is attacking cancer stem cell. So this conjugate is very difficult to develop a synthetic molecule and a natural molecule together, which he has done it through in silico modeling, using the bioinformatics, the AI tools. So now in silico he developed this drug conjugate, and then using organic chemistry, our own synthetic chemistry group, he has developed this conjugate. He has tried it on the in vitro cell lines, in vivo. Now it is ready. They are already completing the clinical trials, and this will go. So here is an example of curcumin, which is known in the natural, the medicine, our own indigenous system of Ayurveda, and then combined with the synthetic molecule of the allopathic system, and then folate. Now he is trying to attack the cancer stem cell. This is an example where the integration is really taking place. Similarly, he has also now developed a Unani medicine, Dawawul Kurkum. Dawawul Kurkum, along with Mariam Sarabat, Biswas, Manoj, we have developed a Unani medicine, which has proved to be very, very beneficial. We looked at the molecular mechanism, safety, efficacy, and now that is for the hepatocellular carcinoma. So we are trying to use those. Now, in fact, we have requested Director Ames. He was here day before yesterday with us on a function, Dr. Srinivas, M. Srinivas.
And we are now trying to look at the partnership with AIMS, that how do we take it to the clinical trials with, we are also trying with ILBS, we are trying with Rajiv Gandhi Cancer Center, and also the APEX organization, All India Institute of Medical Sciences. I'm so grateful that I have two of the experts here. And then this group is also, Dr. Biswas has come out with a very important, the path-breaking research, I would call it, the survival, that how does it lead to the metastasis? Can you predict this? And also this he has done a lot of research, and he made also the presentation over there. I'm sure you will take it forward in this area as well. And then artificial intelligence, I would like to suggest that Amity has established a huge facility, 10 petaflop computing capability today. In the first phase, we are upgrading to 60 petaflop. And I would urge all the experts who have come here for this function, please do visit us. We want partnership. We want that facility to be utilized fully. 2 shifts, 3 shifts. So we are now throwing open, this synergy will come with all the experts, that how do we use this? We are already using it in health. For example, in cancer, that can you predict this early? That can you also diagnose it early? Can you predict the susceptibility using the bioinformatics and a lot of database available today? And also, can you use it for early diagnosis? So we have done it in cancer cervix as one of the important tools. And this will also help in machine learning, deep learning, ultimately leading it for screening. A large number of people in the remote areas, villages, who may not have access to tertiary health care for diagnosis using either mammogram or the cervical cancer to be diagnosed. So, but then if the nursing assistant sitting there in PHC, primary health center, if we can pick up early and then send it to for the tertiary care, that would be great. So here again, the application of AI, ML, DL, integrated with the medicine for screening, like a cytoscan developed by DRDO. So now you can see the cancer cervix, the slides can be seen large number very fast. So this is where it will be like a work hard to screen large number instead of the pathologist looking into hundreds and thousands of slides. The vulnerable, susceptible ones can be seen by the experts. So here again, AI application has tremendous application. Now, one research which I was involved in cancer during my stay at DRDO, I was the DG DRDO for 10 years. So I was also looking after the Institute of Nuclear Medicine and Allied Sciences, where we developed a very important molecule known as 2-deoxy-D-glucose. It's a radiosensitizer. So when you give cancer therapy to the patients, cancer patients, you are not only killing the cancer cell, but you are also killing the normal cell in the vicinity of the cancer cell when you are giving radiation, gamma radiation. So how do you selectively kill the cancer cell? That was a challenge. So what we did, we looked at the cancer metabolism, and the cancer cells are very hyperactive, hungry for energy. And then if you slow down, because they get into glycolytic pathways, and so can you switch off the energy level, which Dr. William Lowry also showed how he is switching off the transporters and also the lactate uptake. So we developed 2-deoxy-D-glucose, which is nothing but analog of glucose. It will be having everything similar to normal glucose, which gives energy and ATP to the cell. But then it can enter the cell, but it cannot give energy. So it cannot enter into glycolytic pathway to produce energy. So when you give this before radiotherapy, in the blood now you have 2 types of glucose, the normal glucose, which can enter the cell and can give energy. The other one is analog glucose, which is 2-deoxy-D-glucose, which can enter the cell but cannot give the energy. So now cancer cell being hyperactive, it is hungry for energy. So it will take relatively more of 2-deoxy-glucose as compared to the normal cell, which will also take it, but then relatively more. So now you can optimize the dosage of radiation in such a way at that level, the sensitized, weaker cancer cell can be killed easily with a lower intensity of radiation. This is how we protect the normal cell in the vicinity, while sensitized cancer cell can be given. Now it went through phase 1, phase 2, phase 3.
And also post-marketing surveillance. Dr. Reddy's laboratory is marketing this. In fact, we repurposed this during COVID to have the same mechanism, switch off the energy to the virus. So same principle we used. We repurposed the drug, same 2-deoxy to save lives of the people. So this was done at DRDO and Dr. Dwarakanath and also Dr. Jain who started this. So this was work where I was busy to facilitate, catalyze this. Now, coming to the final message, the one thing is let's create a consortium, the cancer consortium. I have Dr. Hridesh Prakash sitting here, who's also looking at single repairs and many other methods for the treatment of cancer. He is the center, he has a big center, Cancer for the Center for Translational Research, Amity Center for Translational Research. So this we should create he is organizing a big conference and support affiliated to Cancer Consortium, International Cancer Consortium, and please do participate. Let us build a consortium in research, because otherwise we are islands of excellence, All India Institute of Medical Sciences, UCMS, and many other institutions. But if we come together as one team, as a consortium, then you will be able to move faster, do better, and also come out with more better solutions, whether it's for early diagnosis or for therapy. So let us create a synergy and let's create a consortium, cancer consortium group in the cancer metabolism, which is very important because energy, without energy, whether it's cancer stem cell or it is cancer cell, then you can find many therapeutic needs from this research. So one is consortium. Second is AI. Use this facility. Let us use the facility which we have created. Our Chancellor, Dr. Atul Chauhan himself, has invested here heavily. About 10 crores have been invested to develop this AI center. It's a beautiful center. So I would request, now we have kept the permission for 2 more servers to be added to upgrade from 10 petaflop to 60 petaflop. So use that facility as well. The third one is integrative approach. If you do that, whether it's in the palliative side or an adjuvant, if you can look at what all, like what he's doing with curcumin and what we are trying to do with Dawawul Curcum. So all this is as an adjuvant. You keep the conventional medical management. So wherever you feel traditional systems can play, it cannot replace, the surgical removal and also chemotherapy, radiotherapy, all these are necessary for acute care and the survival rate as well as the lifespan of the people to go up. But then the other part is how do you give the palliative adjuvant care using the traditional systems? This is the integrative medicine. So I would request all the experts and the students, you people keep that in mind when you become leaders. I'm sure you will all take care of this. And by that time, we should find remedies. Before you people get into this, all the experts who are sitting here will be able to bring. I'm so happy that the industry is also here. And certainly, this seminar will throw up many new rules, new path, new avenues, and ultimately leading to the benefit to the cancer patients. And thank you so much for this opportunity to be with you. Thank you.
Thanks a lot, Silvamurthy sir, for his positive thoughts. Now I welcome Dr. Vineet to address the symposium.
Good morning, everyone. So, because we are already running late, so I won't take much time. But the inspiration for this event came when Dr. Rahul called me, maybe last week, and he saw that he will be in Delhi and would like to visit Amity. So then I called Dr. CP, Dr. Mayank, and Dr. Saran, and they're also really like this idea, and I am profoundly grateful for all the speakers that they have accepted our invitation on such a short notice. So it is well known that cancer cell do metabolic reprogramming and to survive in cancer progression. So understanding the molecular mechanism and can lead to identification of different pathways, new molecular targets, and development of new molecular therapeutics. And because these days nothing is beyond or nothing is the beyond the scope of AI. So obviously AI can, I hope AI can definitely help tremendously to identify, to accelerate this research, and will definitely speed up the translation of these research finding into clinical applications. So we are grateful for your participations and enthusiasm. And I believe that PhD student and researcher working in the field of cancer and drug discovery will find this symposium extremely beneficial. And it will offer them valuable insight and provide opportunity to engage with the uh, experts and enable them to exchange idea on the innovation solution in fight against cancer. So thank you for, and I wish productive and inspiring symposium. Thank you. Okay. So now, before we start technical session, let's move for tea. Tea break for only for, so for 10 minutes only. Then we'll assemble back. अभी वेट कर रहे हैं. So we are going to start this session. So please, I request all the audience to come, and we'll start the session now. Okay, so am I audible? Am I audible? Okay, so on the behalf of AMSR, and I welcome all of you for this session. And today we have first speaker in this session, Dr. Rahul Kumar. And Dr. Rahul Kumar currently working as assistant professor in Department of Biotechnology in IIT Hyderabad. And he completed his PhD from CSR Institute of Microbial Technology, Chandigarh. And then for a short stint, he was a postdoctoral fellow in Memorial Sloan Kettering Cancer Center. And after that, he became research officer in Columbia University for 4 years, if I'm wrong. Okay, so in his PhD, he got the best thesis award also. So this is for a PhD student to remember how to do good work. And as you remember in the Selvia Murthy sir message that he was talking about the repurposing of 2G for the cancer and for also for the coronavirus. So today he will talk about Dr. Rahul Kumar on machine learning models and how the repurposing of drug can be used for the better clinical outcomes. So now please join us to invite Dr. Rahul Kumar for his talk. Do you have pointer? Do you have pointer? Laser pointers. It's a cursor. I think laser pointer. Yes, it's one. Yeah, laser pointers. Yes. Yeah, thank you. Hi, am I audible? Yes, I think so. Good morning, everyone. Myself, Rahul Kumar. I'm an assistant professor in Department of Biotechnology, IIT Hyderabad. First of all, I would like to thank Professor Vineet for inviting me for this wonderful symposium. And this is my first time in the MIT campus, actually, and I'm really excited about it. Yeah, so title of my talk is Machine Learning Models from Drug Repurposing to Predicting Clinical Outcomes. So, machine learning, the application of machine learning in biology and life sciences is not very new to this audience, and we know a lot of applications are being done, using machine learning, and a lot of problems being solved in the field of medicine and the life sciences. So our lab at IIT Hyderabad also work in the same domain. We use machine learning algorithms to solve multiple biological problem using big data sets in genomics, transcriptomics, and large in vitro screens. And drug repurposing is already introduced by the Dr. Murthy in the morning, so that make my job very easy actually. So today in this talk, I will be presenting two of our recent project which we have done in our lab at IIT Hyderabad. So first project is QSAR based drug repurposing, a novel approach for colorectal cancer. So if we talk about drug repurposing, so drug repurposing is a very important aspect of drug development these days in the pharmaceutical industry, where we try to look for the new uses of existing drugs actually. So if you look at this figure, where we are comparing de novo drug discovery process and the drug repurposing. So as you can see in the de novo drug repurposing, it takes around 10 to 17 years, as you all know. But in case of drug repurposing, it takes just 3 to 12 years, because we can bypass, very important things like ADMET and lead identification, which, kind of speed up the whole process of drug discovery actually. And if you look at this figure at the right, so if you see, we are comparing here risk versus reward. And if you see the de novo drug discovery process, the risk is very high. Obviously, the reward is also high, but the risk is very high. But as compared to the de novo, the drug repurposing is basically having the low risk, but the reward is as high as de novo actually. So that's why this drug repurposing concept is very important these days, which we can speed up the drug discovery process actually. And so if we talk about the drug repurposing approach, so this figure is compiling those approaches which we take to do drug repurposing. So this figure, we have 8 approaches of drug repurposing actually. So in the blue, we have computational approaches. So as you can imagine, so computational approaches is very important in the drug discovery process these days. And 6 out of 8 is basically computational approaches, and 2 are experimental approaches. So, and the table you see here, the right side, these are some of the repurposed drug, which we repurposed for some other diseases actually. So this list is long. I just took few examples here for this presentation purpose. And what we are interested in drug repurposing is this particular aspect where we have large screens, large in vitro screens, and plus combined with the genomics technology actually. So we came up with these two databases. One is the genomics of drug sensitivity in cancer. Okay, so these guys from the Sanger Institute in UK and the MGH from US, they screen 1,000 human cancer cell lines across various cancer types, and they screen around more than 100 100 drugs on these cancer cell lines actually, and the data is publicly available on their website. And the Cosmic is very well known in the field of cancer actually, so where they have characterized again the more than 1,000 cell lines, genomic characterization, transcriptomic characterization, and all the data is publicly available to use. So we thought of using this data to do some drug repurposing actually. So we worked on the colorectal cancer. So we in this database we had around 46 colorectal cancer cell line, and they screen around 297 drugs on these 46 colorectal cancer cell lines, and roughly we had around 11,000 drug cell line combination in this data set actually. And so if we talk about how we can use this data set, to do some drug repurposing or develop some machine learning based models. So if we talk about traditional approach, which is QSR, quantitative structure activity relationship, and we talk about QSR where we generally have, a large chemical screen library, and we have, a target, and these molecules are basically screened on this binding target, which is generally a protein target, and we have data in terms of affinity of these molecule against this protein target, and we develop various machine learning models. But the problem with us here was we did not have a protein target actually. So what we had was we had 46 colorectal cancer cell lines on which 297 drugs were screened. We did not really have any protein target, so that was a challenge. So we thought how to go forward, because we do not have any protein target actually. So we thought of developing cell line based QSR models instead of models which are specific to a particular protein target actually. And so we had IC50 value of all these drugs against these cell lines, and we calculated the chemical descriptors using computational tools, and we did machine learning. We applied different algorithm, and we got good results using SVM, which is support vector machine. And using this data set, we developed cell line based QSR models actually. This is non-traditional QSR models, which is traditional models generally against a protein, but this is against a cell line actually. Right. So this was the overall workflow of this work we had done actually. So we, as I said, we had around 11,000 drug cell line combinations, and we filtered out some of the drugs which was not very efficacious, and we took the SDF files of these drugs and calculated the chemical descriptor using a parallel software, and we did the feature selection and selected the best descriptors which we can go ahead for developing our QSR models using we use tenfold cross validation technique which is very well known in the field of machine learning. And we tested our QSR model using test dataset as well as the independent dataset, and we did some further downstream analysis which I'll talk about. We look for the descriptor analysis, we look for the drug to oncogene analysis, and we also did some drug repurposing, which is the main object of this particular study actually. So if we come to the model performance, so these two tables are showing the performance of our model. So we had 46 colorectal cancer cell lines, and we developed models against all of them, but we found we could able to develop only these good models against only the 12 colorectal cancer cell lines actually. So we had accuracy in terms of R square, R, RMSE, and ME. As you can see, we achieved all R square of 0.6 in almost every cell line actually, and very less RMSE and mean absolute error, less error and high accuracy. And this is on the training data sets, and in machine learning we always have to kind of validate our models on the test data set as well. So we also got more or less the same kind of accuracy in our test data set as well. So this matrix is showing that we got a good model against at least 12 colorectal cancer cell lines. We started with 46, but we got only for the 12 cancer cell lines actually. So what we did, what we're doing here, we basically try to add another layer of validation of our QSR model actually. So what we took this gene ABCA13, which is kind of mutated in around half of the colorectal cancer cell line, and as you can see here, the mutated cell lines have less potency against this particular drug tramitinib, which is a kinase inhibitor, using the actual log IC50, which is identified using experiments, and using the predicted IC50 as well, we kind of recapitulated the same trend. And again, the gene FSIP2 and this drug IgG 946, which is basically methyl transferase inhibitor. So here we were seeing that the mutated cell lines were kind of more susceptible for this particular drug using the actual log IC50, and in the predicted log IC50 also we kind of recapitulated the same trend. So which also tells that the model we have developed is kind of able to recapitulate the biological phenomena here actually. And so we were also interested to know what kind of chemical descriptors, impacting the biological activity of these drugs. So we had around 18,000 chemical descriptors actually, and we were trying to look for, the descriptors which are really impacting those, kind of affecting the impact, increasing the activity of the drugs, for example. So here we have, we found two chemical descriptors, which is FP3, fingerprint 3, and graph 252. So one is basically the presence of that particular chemical descriptor in these drugs. So one dot here is basically a drug actually. So if you have this chemical descriptor present in these drugs, so the IC50 is low, means the drug is more efficacious if these fingerprints are present, and if they're absent, so the efficacy is not that good in case of these 4 colorectal cancer cell line we have in this particular study here. Yeah, so the next main goal of this study was to repurpose the FDA approved drug actually. So what we did, we took around 1627 FDA approved drugs from drug bank, and we screened these FDA approved drugs and predicted their activity using our cell line based QSR model, which we have just developed. And then we looked at the top 11 predicted drugs, and interestingly we found that out of 9, the most of them were already known, well, either they are anti-cancer in nature, and they are already discovered. People have discovered their anti-cancer activity actually. So what we found, that 2 drugs, one is viomycin, which is antimicrobial, and another is diamorphin, which is analgesic. So these 2 drugs, which we are predicting as having some anti-colorectal cancer activity, but they are not yet been tested. So in the experiments, we would like to take this drug forward to check whether they are really having some anti-cancer activity or not. Yeah, so to summarize this work, so we collected a large in vitro screens from GDSC2 database and filtered out some of the drugs, and we calculate and selected chemical descriptor and develop our machine learning based QSR models, and then we analyze some descriptor analysis to oncogene and target and path, which I did not show here. And we also found 2 drug, viomycin and diamorphin, which yet to be validated using experimental technique actually. And what we did, we integrated these models into web server form, which is called as Thread, and where user can come and just check if they want to check any anti-cancer activity in their compound against this colorectal cancer cell line. So this is the web server interface which we have developed. We integrate all those 12 models in this web server, and this is publicly available at this website, and users can use this website to predict anti-colorectal cancer activity for their unknown compound if they wish. Yeah. So now I would like to switch a gear to another project which we had done in a lab. So where we basically use deep learning methods to classify the molecular subtypes of diffuse large B-cell lymphoma. So diffuse large B-cell lymphoma is a very is a type of a blood cancer, and we use this model for the predicting the clinical outcome, which I will be talking about. So before going into our work, I would like to just briefly talk about this a little bit of introduction. So DLBCL, diffuse large B-cell lymphoma, is of mainly 2 types, GCB and ABC. So which originate from the germinal center B cells actually, and germinal center B cells have 2 zones, which is one is the dark zone where the somatic hypermutation occur, and the light zone where the memory B cell form actually. And so there are 2 major subtypes, molecular subtypes, GCB and ABC, and they have very unique pattern if you look at the transcriptomic data, so they have a unique pattern, different, like for example, MYC only present in the dark zone, and it's totally absent from the light zone. So they're very well characterized, and this study, these researchers have found that the activated activated B cell, like, which is ACB subtype of DLBCL, is the patient belong to this particular category having a poor survival actually, and the GCB they have little better survival as compared to the ACB like DLBCL. So but the shortcoming of this study was this study was just based on 40 patient, which is kind of very less, but now due to the genomic revolution, we have a lot of data, transcriptome profiles are available. So, for example, we had these two profiles where the researchers from this group, they did a transcriptome profile of 381 DLBCL cases, and the researcher from this group, they screen around 294 DLBCL cases. All are clinical, all were the clinical data patients, and so this is a huge data set. So we thought of developing some classifier which is not been there yet. So using the data, so can we classify these activated B cell like and GCB like DLBCL, which clinician can use in the clinics? Right. Oh, so we planned, we devised a workflow for this study using machine learning algorithms. So what we did, we took this data set as a training data set, and this NECATL we took as testing data set actually. So we're starting with the DLBCL cases, 381 DLBCL cases. So we divided them into training data set and the testing data, which is kind of traditional thing we do in the machine learning models. And what we did, we found, because we had the gene expression data of around 18,000 genes actually. So all genes are not important, from the biological perspective. So we kind of filter out, we kind of like do feature selection using information gain algorithm. We found top 20 genes which are highly discriminatory in nature to classify between ABC and GCB DLBCL patients. And we did tenfold cross validation using our training data set and developed this model which we named as MLP CEO. So MLP is machine multi-layer perceptron, which is a type of a deep learning method, and COO is basically a cell line of origin. It means that we're just telling you the molecular subtypes of the DLBCL patients actually. And then we tested this model, using testing data set as well as the independent data set. Yeah. So before going to the performance of these model, what we did, we developed this heat map, and where we look for the these top 20 genes, how their expression patterns are behaving in training and the independent data set. So as you can see here, we have two subtypes. Green is GCB. This is phenobar showing the GCB and ABC subtype. This is green is GCB, and the red is ABC. So as you can see, the transcriptomic pattern is really discriminatory in nature for these 20 genes actually, and both in training as well as the independent data set. So we also did this PC analysis just to reconfirm that what we are going ahead with these 20 genes, whether they are really discriminatory in nature for these two subtypes or not. So again, we find, as you can see in these two clusters, red is ABC, green is GCB. So these the transcriptomic profile of these 20 genes are quite discriminatory in nature for these two molecular subtype of DLBCL. Yeah, so coming to the performance of these models, so we achieved a good accuracy, mostly more than 0.95, and ROC also around 0.95, as you can see here. So we and in both in all three data set, training, testing, and independent data sets. So it's reconfirmed that the model we have developed using these 20 genes performing very well and can be used further. Yeah, so what we did actually at the end, we took these our models and tried to see whether they can be extrapolated to some clinical outcome or not. So here you can see this, the top two panel is from the training data set, and if you see using the actual CEO, which is identified using the pathologist, and you can see the survival data where ABC are having a poor survival as compared to the GCB. This is using actual cell line, this molecular subtypes or cell of origin, and in the predicted, which we predicted using our models, they also kind of recapitulating what we know using the experiments. So it's both in training as well as the independent data set. We kind of recapitulating the survival outcomes of the patients using our machine learning based models. Yeah, so this is my last slide. So I would like to acknowledge my PhD students, Kavita and Avik, who did most of this work. And I would like to thanks our alumni and collaborators and the funding agency for this work, actually. Thank you so much. Thank you. Thank you, Dr. Rahul, for sharing such a nice information with us. And little bit, one question I want to ask that today people are looking for drugs at the nanomolar level, but in your first story, you put the filter at the 100 macromolar. What is the reason behind that?
Yeah, so the reason for that is to remove the drugs which are not efficacious actually. So we remove drugs having more than 100 micromolar IC50. So if you have IC50 or 100 micromolar, it means drug is not working. So we remove those drugs actually. We did not use those drugs in our machine learning models actually.
Can I ask one question?
A few questions, yes.
I'm AK Singh. I think very wonderful talk. Very wonderful talk. I appreciate. In your first part of repurposing, where you said that you have found a newer use of vancomycin and diamorphine as a anti-cancer, though they were projected as antibacterial. So just for my information, did you screen the literature to find, was there any mention by any researcher that reporting their anti-cancer activity? Because my intention is then where did we lose it?
Yes, it's a very good question. So it also came to our mind, and we did all thorough literature search, and we couldn't find any known anti-cancer activity for these two particular drugs. We did for all the top 10, 11 drugs actually. We found for some of them, but for these two drugs, we did not find any known, activity in the literature actually. So that's why we projected them that they could be taken forward for the experimental validation actually.
So I think you would give credit because QSAR analysis is not new, but I think your AI in understanding interaction of drug at gene level or influence of the drug at gene level is giving you this new insight. I think we would be more interested to see your last experiment, that how does your predictive model, the information you got from your models, how do they translate into the clinics?
Yeah. Definitely. We are also looking forward to this actually, and we are looking for experimental collaborator for doing this. Yeah, definitely. Yeah, very nice suggestion. Thank you so much.
But wonderful. My congratulations to you. Wonderful talk. I enjoyed listening to you.
Thank you.
So we can take question from students. No. Okay. So I request. Okay, Dr. Munaj.
So, so as I said, we had around 46 colorectal cancer cell lines, and against which 297 drugs, roughly 297. So those 297 were not tried against all 46, but it's a range. Let's say 100 to
No. In general, how many data set is required to have any model to predict?
So always it is as much as it is possible, but that is always not possible. So I think what we for machine learning, I would suggest at least 50 plus we should have actually. So
Thank you.
Hi Rahul, it's a wonderful talk. My question is that you talk about vancomycin, right?
Yes.
Did you start wet lab work on this drug or?
Yeah, so we wrote a grant actually to ICMR for this, and we're still waiting for funding to validate these drugs. Actually, we wrote a grant with our experimental collaborator in IIT Hyderabad in our department, but we are waiting for funds to do some experiments. Actually, we definitely will do the experiments. Yeah.
In my opinion, it's an antibacterial drug, right?
Yeah, right.
So the colorectal might be many of the bacteria, especially H. pylori or other bacteria. Now, it has been very well known that they contribute to the carcinogenesis. Right. Apart from the mutation, they accelerate the rate. So, in my opinion, if you look at the microbiome with this drug in relation to cancer, will be a addition to the anti-cancerous drug, which are people following nowadays.
Yes, very nice session, Dr. Manoj. Yeah, we'll take this forward. We did not do this study, but yes, we can try that because it's a antibacterial drug. We should look at from the angle of the microbiome as well. Yeah, thank you so much. Yeah.
Hi. Hello. Hi. This side. Yeah. So my question is from the first talk that without clear knowledge of your target, what guided you to select the compounds for QSAR studies?
Yeah, so that's what we're wondering about, because we did not have any protein target here. What we had is just biological activity of a particular drug against the whole cell line, and where we know the target of some drugs, but not all. So what we actually in this study, we're trying to look for some chemical descriptors, a general chemistry, chemical nature of a particular drug, which can could have some activity against colorectal cancer cell lines. So as I said, it's a non-traditional QSR model where we don't have protein target. So to nail that down, we did some genomic analysis, which I showed as well. But yeah, we did not have target against. Yeah.
So you selected compound randomly or like
so not randomly. What we selected any drug which would try against the cell line and have IC50 more than 100 micromolar. That was the cutoff. We did not screen for particular pathway, particular target. Yes.
Okay,
Hello. Can I have a question? Rahul, it's a wonderful talk.
Thank you.
And it's a wonderful talk. It's not a question. I want to know, with the use of AI and ML, as you screen a lot of drug in your model, so can we have an idea about, okay, this functional group in a drug may improve the cell line activity or binding affinity? Can we develop a model or something like, okay, this kind of entity is active, and if we add these type of functional group, it may enhance the or improve the activity? Can we do that?
Yes, yes. So thank you so much for asking this great question. So actually, we did that. We tried to. So as I said, we calculated around 20,000 chemical descriptors and selected them based on the activity, and we found two chemical descriptors, FP3, which basically are aromatic ring for attached with the aldehyde group and the the carboxylic acid group. So these kind of entity we found, but because we are not the chemist, we cannot take them forward. But we did find these chemical descriptors which might be imparting the real anti-cancer activities. So yeah, this is one aspect, important aspect which we can take with the help of chemists. But we, as a biologist, could not took this forward. But yes, we did find some chemical moieties in these drugs actually. Thank you so much. Yeah. So now I will invite Dr. Biswas sir, Director of AMSR, for the felicitation.
Please come. आ जाइए, आ जाइए. So now we will move to the next speaker. So next speaker is Dr. Chandra Prakash Prasad, who has received his PhD from School of Medical and Paramedical Health Science, Guru Gobind Singh University in Rasta. And then he has a huge experience from Lund University, Sweden. And currently he is working as an additional professor at the Department of Medical Oncology, All India Institute of Medical Science, New Delhi, India. Dr. Prasad has published various papers in well-reputed international journal, and he has several awards in his account, both nationally and internationally. I invite you to listen your talk. Looking forward. Thank you.
afternoon. First of all, I want to thank the organizer, especially Professor Vineet, for inviting me here and to discuss about our research. So today I will be speaking about aerobic glycolysis in TNBC, the role of beta-catenin, PFKP, LDHA, MCT1 axis as theranostic target. So in the morning, we heard Professor William, and he was talking about aerobic glycolysis, lactate. So I'll go in deep with my particular set of disease, that is breast cancer, which I'm working on. So if you look at breast cancer, just like worldwide scenario, it is most common cancer in India. And if you look at ER negativity, which is in West around 15%, in India, we have around 25 to 35%. And we have a subset known as triple negative breast cancer, where the patient harbor no estrogen receptor, progesterone receptor, or HER2 receptor. So we don't have any therapy to target, And if you look at these TNBCs, they are really aggressive cancer. And as I told you that there are no target for the cancer, chemotherapy is the mainstay, and most of the patients are treated with chemotherapy. And if you look, these patients, around 50 to 70%, patient come to the clinic again because of the relapsed cancer. So my lab is working in the area of cancer metabolism, specifically aerobic glycolysis, where we are looking for the markers if TNBCs can actually differentially express any marker of aerobic glycolysis that we can actually inhibit. So just to summarize what aerobic glycolysis is, if you look at normal cells as shown here, in the presence of oxygen, glucose is mainly converted to pyruvate, and this pyruvate mainly goes to mitochondria for the energy generation. We call this process oxidative phosphorylation. However, in tumor cells, irrespective oxygen is there or not, pyruvate is mainly converted to lactate. Now, this lactate, when it comes out into tumor microenvironment, what it does is that it suppresses immune suppression, it increases the metastasis, therapy resistance, and of course, it acidifies the tumor microenvironment, so making metalloproteases active, so tumor actually invade much faster. So while working in my postdoc, I was working on Wnt5a signaling. So if you can see the signaling here, Wnt5a are ligands that are secreted by the cells. They bind to their cognate receptors and activate the signaling. What we found that in breast cancer cells and TNBCs, when you see there is a loss of Wnt5a, in both the cases, breast cancer as well as in TNBC, the patient shows less survival, making this Wnt5a a tumor suppressor. So if it is there in normal, it is basically fighting tumors. But when it gets downregulated, you get an onset of tumor. So what we found that while working in cell culture lab, where we are growing TNBC cells, we found that these cells are negative for Wnt5a. So when we gave the Wnt5a from the top, which is recombinant Wnt5a, or we overexpress Wnt5a in these cells, we found that these cells actually show less migration and invasion. So one of the aspects that we looked after that, whether Wnt5a can actually alter the metabolism. So what we did, we took the overexpressing Wnt5a cells where Wnt5a is overexpressed, and we actually looked at glycolytic markers. We found a protein, PFKP. It is isoform of PFK1, phosphofructokinase 1, which we found is downregulated. And the same cells actually show less lactate. So we thought maybe Wnt5a is inhibiting this PFKP, thereby inhibiting the aerobic glycolysis and the lactate production. If you look at PFKP, it is one of the isoform of PFK1, that is phosphofructose kinase 1. And it basically has three isoforms, and of the three, only PFKP, that is platelet isoform, not the liver, not the muscle, was associated with the bad prognosis if it is overexpressed in the breast cancer patients. We further show that if you knock down PFKP, you knock down lactate. So basically what is happening that Wnt5a, when there is a loss of Wnt5a, this PFKP is getting up in cancer patient, doing all the bad work, upregulating the aerobic glycolysis and lactate. So when you give Wnt5a back to these cells, it is inhibiting aerobic glycolysis by inhibition of PFKP. We further show that beta-catenin, which is one of the hallmark of Wnt signaling, is actually sitting upstream to PFKP, because when we inhibit it with beta-catenin inhibitor XAV, we found there is a inhibition of PFKP in both the TNBC cell lines. We further show that if you knock down beta-catenin, which is upstream of PFKP, basically you get less lactate. So beta-catenin and PFKP are in axis, which is actually getting upregulated by the downregulation of Wnt5a. Now, till now, we are sure that, okay, beta-catenin and PFKP are regulating lactate in TNBC cells. Now, this lactate is coming out from these cells. So, can Wnt5a also mask these cells if we overexpress Wnt5a from the lactate, which is in the tumor microenvironment? So, we show that here, if you look, these are Wnt5a overexpressing cells. These are empty vector transfected, and these are Wnt5a transfected cells. When we gave them lactate, you see there is migration, there is an increase in migration. However, when Wnt5a is there, even if you give lactate from outside, there is no increase in the migration. And we found that it is because Wnt5a inhibit a channel known as MCT1 that actually stops the intake of lactate in the cancer cells. So we come up with this pathway that Wnt5a, which is a tumor suppressor, if you give Wnt5a in tumor cells back, like TNBC cells, it is inhibiting beta-catenin, PFKP, and the lactate production. It is also inhibiting MCT1, so the extracellular lactate can't come inside the cancer cells. On the basis of this, we actually made a peptide known as FOXY5, which just worked like Wnt5a, because you can't give recombinant protein to patient. So we developed this hexapeptide, and we have shown that it works in the same way, just like Wnt5a, and it is now in phase 2 trial for breast and colon cancer. So that was the story we published in 2017, that we are close in finding like a new pathway where Wnt5a is regulating aerobic glycolysis. Now, one of the important enzyme is LDHA, which is actually converting pyruvate to lactate. So we, in next study, we want to see whether LDHA is also part of this pathway. So what we did, we knocked down PFKP and looked at LDHA, which is downstream of PFKP. As shown here, when you knock down with siRNA of PFKP, there is a reduction in LDHA also, and the reduction in lactate, thereby setting up a pathway. Yes, beta-catenin is upstream, then PFKP, then LDHA, and then it is regulating lactate. We further show that there is a nutraceutical compound quercetin, which actually inhibit this axis, PFKP, LDHA, as shown here. It's an immunofluorescent. The green color is actually PFK protein. So when you give quercetin to it, you see there is a downregulation in the green part. Basically, there is a reduction in PFKP protein, as shown in the Western blot. And yes, quercetin indeed inhibit LDHA also and lactate. So now, in TNBC, we have like a pathway where beta-catenin is regulating PFKP, PFKP is regulating LDHA, which is actually involved in the production of lactate. But these are cells. Is it really happening in TNBC patients also? So we looked into patients by immunohistochemistry. We looked all the 4 proteins, beta-catenin, PFKP, LDHA, MCT1. In 98 patients, it's a cohort that we have prepared in the All India Institute of Medical Sciences. And we found that beta-catenin expression is overexpressed in 64% of TNBC patients. And all the other 3 proteins also are overexpressed in more than 50% of TNBC patients. We also found that beta-catenin is positively associated with PFKP expression, as shown here, as well as MCT1 expression. Now, of these 98, we followed 71 patients, and we carried out Kaplan-Meier analysis just to see whether the overexpression of these pathway proteins can actually associate with the less survival, because it actually signifies the aerobic glycolysis. To our surprise, not the single protein, either PFKP or beta-catenin or LDHA, MCT1 are associated with the survival. But when you combine these, basically you are getting a better picture to the survival. When they are overexpressed, the survival is less. And the best result was achieved when all the 4 markers were combined. As seen here, that patients, so of the around 71 patients we found, around 30 patients were actually having these all the four protein positivity, and we found the survival was really less, thereby suggesting that this pathway is active not only in cell lines but in patients also. And inhibiting this pathway, basically you can give us results, by inhibiting aerobic glycolysis, thereby inhibiting the migration and progression of cancer cells. So what we did, we then went back to cell lines and started with a XAV939, which is a known beta-catenin inhibitor. So I have told now it's a pathway. So if you inhibit upstream, all the 3 in the pathway should also get downregulated. So what we show here is that if you use XAV939, inhibit beta-catenin, basically you get the downregulation of all the 4 proteins, and we have tested 2 breast cancer cell lines. Also, we have shown that there is a reduction in lactate. But to our surprise, the lactate reduction was not that much, and we thought of increasing the concentration. But at the same time, we got some clinical trial reports in 2020 that XAV939 has acute side effects. You know why? Because XAV939 is a beta-catenin inhibitor. Beta-catenin is a very important protein for the normal physiology also. So basically, you are affecting normal cells also. And most of this toxicity was associated with the colon. So now, at this time, we thought of using a much safer drug. So we came to know about this drug, exitinib, which is a VEGFR inhibitor, tyrosine kinase inhibitor. And we found that exitinib also does the same thing, what XAV939 does. And it actually affects aerobic glycolysis much better than XAV939. The good thing about exitinib is it's already FDA approved for renal cell carcinomas with a manageable toxicities. So what we need to do is that we need to try this into TNBC patients. So we took exitinib further and we looked into, this is a cell invasion assay that we have developed in the lab. So what we are doing is that we are making a droplet of the cell, and you can see that there are 2 cell lines. This is MDMB2T1, 468. We treated with XAV939. This is with exitinib. These are the control cells, and with the spheres, you can see with time the cells will invade out. So compared to control, you see less cells in 231 treated with XAV939, similar in 468 also, as shown here in the graph. But in exitinib, the effects are much better compared to XAV939. We went further to do animal experiment, and we have shown that with exitinib, basically you can inhibit 41 xenograft model, which is the TNBC cells, like in a really big way. And as shown here, as compared to control, where you can see the dense tumor, you have like a perforated tumors in exitinib treated cells. Moreover, we have shown the beta-catenin expression is also getting downregulated with AXAB treatment. So that's one part of the story. Now we are done with the preclinical trial, and we are thinking of running a clinical trial, because I'm working in medical oncology department, so we have like clinical counterpart also. So I've shown the data, and they are convinced, and they want to run a phase 2 clinical trial with exitinib on TNBC patients. This is another story which I like to share. We have published recently that there is a concept that in tumor microenvironment there are cells which can produce lactate, and which we call glycolytic cells, or showing high aerobic glycolysis. And there are another cell type, basically, which we call at oxidative tumor cells. They actually take this lactate, convert this lactate to pyruvate, and this pyruvate is used by oxidative phosphorylation for energy generation. So actually this work was we were doing in oral cancer cells. So what we were doing is that we want to see the effect of lactate on oral cancer cell lines. So what we did, we treated with lactate salt for like 2 months, and we found that cells after the 2 months of treatment with lactate are showing mesenchymal characteristics. We further show that if you give a short-term treatment also, like of 48 hours, you can see similar effects in these cells. We have shown that the similar effect was also seen in TNBC cells when they are exposed to lactate for 48 hours. And both oral cancer as well as TNBC cells shows that when you give them lactate, they show increased migration. Now, in the morning, we were listening to William, and then he was using this drug AZD3965, which is the MCT1 inhibitor, and then he told us that it doesn't work alone. You need to inhibit GLS1 also. So that's a new learning today, because we also find that this drug is not working alone. When we give MCT1 inhibitor in the presence of lactate, we didn't find any change in mesenchymal marker. But when we gave a drug known as 7-ACE2, which is a MPC1 inhibitor, there is a significant decrease in the mesenchymal marker for both oral cancer as well as TNBC cells. So what this 7-ACE do? When you have lactate in the microenvironment, this lactate come inside the cytoplasm, converted to pyruvate, and this pyruvate moves to mitochondria. For moving this pyruvate to mitochondria, we need these channels, mitochondrial pyruvate channel 1 and 2. 7-ACE inhibit this. So we also show that 7-ACE inhibits the migration in the presence of lactate. And what we found for the first time is that when you give lactate actually to these cells, either oral or breast cancer cells, with increasing concentration of lactate, they basically increase MPC1 expression also. And that's why when you are giving 7-ACE2, the effect of increased lactate is basically going away. So this is the model. This is what we are suggesting, that in oxidative cancer cells, when the lactate is outside, basically you can inhibit the invasion and migration of these cells by using 7-ACE2, but not with the MCT1 inhibitor. So to conclude, our present investigation suggests that employing inhibitors targeting the beta-catenin, PFKP, and MPC1 could be a promising therapeutic strategy to effectively manage triple-negative breast cancer cells. So these are some of our reviews that we recently published and the papers, if you want to go and study these. And most of the work of Exidin was done by Umar, who is my senior PhD student. And the 7-ACE work was done by Akansha. She's a third-year PhD student. And I want to thank the funding, ICMR, DST, and AIMS. Thank you.
Hello, CP. Yeah, this is my question is from the first story.
Yes.
Because you have shown that is Win5A.
Win5A, yes.
Yeah. So after that, you have shown some of the survival cut along with the 4 markers, including the NCP1, right?
Yes.
So in that survival curve and based on the relation, what is the status of Win5A that was missing in your data?
Yes.
And yeah. Second thing is that in the Win5A,
yes.
I have found in their western blot there is a two bands.
Yes.
So what are those?
Yes.
Yeah.
First thing, I'm coming with the western blot.
Yeah, okay? Right.
So detecting Win5A is a problem.
Yes.
Okay.
Of course.
Yes.
Yeah.
And in my postdoc lab, we were working on Win5A. So we tested around 20 odd antibodies. Mm-hmm. And this is the band that we got with R&D antibody. There are 2 bands. But when you knock down with siRNA, the band,
upper bands, yes,
is the Win5A.
Okay.
So even if you actually, what do you say, in the lysate, like TNBC cells which don't express Win5A, if you overexpress Win5A, they also give you these 2 bands when you actually probe with R&D antibody. Okay. But the upper band is Win5A.
Okay.
Now coming to the story number 2.
Yes.
Win5A was just a start. Okay.
Right.
So
right, of course.
I am a signaling guy. So in my PhD, I work on canonical signaling.
Yeah.
And in my postdoc, I work on non-canonical. So canonical is beta-catenin independent.
Of course.
Non-canonical is independent. But now stories are coming that it is actually through canonical. So what we can actually suggest that in breast cancer, what is happening that in normal, the non-canonical is up.
Yes.
But when it moves to cancers, non-canonical goes up and when non-canonical is down. So Win5A is down in cancer cells. And because of which beta-catenin is getting overexpressed.
Right.
So when we overexpress Wnt5a, we saw there is an inhibition of beta catenin and PFKP and then further downstream. So idea was in TNBC cells, then we left Wnt5a from there. Whether patients also show the same thing. Do we have beta catenin, PFKP, LDHA, MCT1 overexpression in these cells? Patients.
Yeah. No, my question is on regarding the tumor microenvironment.
Okay.
If you think about this as marker and you have some kind of the database with this 4-1.
Yes.
So any database is available.
We have not looked in our database about Wnt5a protein.
I see.
Because now we have focused mainly on the canonical part. So I have left the non-canonical part there.
Okay.
But it can be done. But we know that in TNBC, there is a loss of Wnt5a. It's nothing to.
Again, Dr. Viswas, we can discuss if during the live
questions.
Now I will ask if there is any question from the back.
Students.
Any question? Yeah, please. Very good.
Good afternoon, sir. The presentation was quite nice. I have some question. Like as I mentioned that epoxy 5 is a substitute of Wnt5a. So it is how much it is effective in fighting tumor. And like another question is, what if the epoxy 5 is down regulated? What will it cause to the BRCA1 cells?
Okay. So if I get your question right, first thing, FOXY5 is a hexapeptide that mimic Wnt5a. As I told you that Wnt5a is a chemosuppressor in breast cancer. So how to actually supplement these breast cancer patients with Wnt5a? Of course, you can't give recombinant Wnt5a protein because it's really big. So we developed a hexapeptide. Basically, it's a 6 amino acid sequence that mimics like Wnt5a. So of course, because it is not a 360 amino acid long Wnt5a, it only does few things like Wnt5a, like it activates calcium signaling, it activates PKC, it inhibits the migration and invasion. This is what we have actually shown in various publications before actually going into the clinical trials. Now, FOXY5 is in phase 2 in Europe, mainly in Belgium and Denmark hospitals, where we are actually looking at the effect of FOXY5, where Wnt5a is like a tumor suppressor, cancers like breast cancer and colon cancer, the effect, right? That's the first thing. Now, FOXY5, we have not seen its effect on BRCA-ness, but let it come like after phase 2, maybe we can see the results and we can try that, or we can try in combining with like, if you are referring to PARP inhibitor or something like that. We are open to do that, but still we are still waiting for the result from phase 2.
Thank you. Any question from the front? I think there is no more question. Thank you very much for your interesting talk. Now I will hand over to Priyanka to carry forward.
So thank you, Dr. Chand Prakash, so much for your insightful talk. And I would like to request Dr. Abneet and Dr. Manoj Garg to facilitate Dr. Chand Prakash. So for the next talk, I would like to invite Dr. Reshma Rani, which she will speak on drug discovery, and ma'am is from Jubilant Biosys, Noida. So I welcome Dr. Reshma.
Yeah, well, it's my pleasure to introduce Dr. Reshma, who is leading a team of scientists in Jubilant Devices in the field of pharmaceuticals uh, and also medicinal chemistry. She has done her PhD and masters in IIT Roorkee, and she went for postdoc uh, in Osaka University, Japan. And she also got the Marie Curie Fellowship, and she worked in Italy also, in the University of Pisa, I think. So she has published many publications, around more than 50 publications she has. And her interest is cancer metabolism and metabolic inhibitors. And now I request Dr. Resma to continue.
Thank you, Dr. Prakash, for a nice introduction, and good afternoon, everyone. So I will not talk much about the biology because I am not biologist, and I will talk in during my talk. I will try to discuss about the drug discovery and the molecule inside the body, what the challenges a medicinal chemist face to discover a medicine. It's not a just a thought. We just discovered a molecule and we had a medicine. It's a hard work of not a medicinal chemist. It's a hard work of computational chemist. It's a hard work of chemist, medicinal chemist, you can say. It's a hard work of biologist. And then the DMPK people. Means this is the collectively effort. If we remove one effort, we cannot do anything. So everyone is important for to translate a molecule into a market as a drug in a clinic. So here I will discuss a little bit about tumor glycolysis, and I will give us some clue how we can design a molecule by targeting a enzyme or the protein or the micromolecule. So first, in the morning, William Laurie discuss about the energy, and then Dr. Silva Murthy discuss about the energy, and then again Chandra Prakash talk about the energy. So and everyone, we came here in MIT University by consumption of energy in the form of vehicle. So engine in your car, everything is fascinating, light, lamp, color, everything. But the hardcore thing is engine, energy. If you are not feeling your car, your engine in the car, what happened? The car will stop. Similarly here, everyone needs energy. We needs energy to do everything, whatever we are doing. We are reading, we are eating, we are. It means it. If I am seeing this, I need the energy. We need the energy. So energy is the ultimate role of our body. Similarly, the cell needs the energy, and we are talking about the cancer cell. And everyone knows, you guys knows, the BSc and MSc, every student knows about the cancer cell. Means cancer cell is the fast growing. So if they are fast growing, what they need? The energy. So here, this I'm discussing about the simple cell. So glycolysis is the main source where glucose is the main source for glycolysis, where we are using where the cells producing energy, where we the body needs. So what is the glucose use this oxidative phosphorylation and produces 30 to 36 ATP. That is the combination effect of glycolysis plus oxidative phosphorylation in TCA cycle. Then this is the normal situation when the cells in normoxic condition. We have sufficient oxygen. Suppose we don't have sufficient oxygen, the cells in hypoxic nature, but they are normal cells. So what happens when you are doing the exercise? Then in that case, your cells are in hypoxic in nature a little bit. Then in that case, the glucose is not converting the exact amount of ATPs. They are producing less quantity of ATP, and this is the scenario when a cell is in hypoxic nature, then a per glucose molecule, that only 2 ATP is forming. Once the cells is in hypoxic in nature. So this is the anaerobic glycolysis. And you know, this is the glycolytic phenotype the cancer cells are following. And cancer cells are very clever. They adopt the glycolytic phenotype. If you block any enzyme, then the William Laurie says they adopt another mechanism. So it is a challenge how to target a enzyme. So now this fact, the glycolytic phenotype is not a new fact, new hypothesis. So it was discovered in 1931 by German scientist, and he got the Nobel Prize for that. And this concept was this concept is utilizing in clinic for FDG imaging, where this is the fluoro-labeled glucose. There is a small unit fluorine that create the imaging here. So the Otto Barber hypothesized that the cancer cells are hungry for the glucose, and glucose are the main source for the energy. The cancer cell consume glucose and produce the lactate in excess. If we compare with the normal cells with the cancer cell, what happened? The glucose consumption by the cancer cell is almost 80 to 90% in comparison to the normal cells. So lactase is in excess. If the lactate is in excess and lactate is in acidic in nature, so what happened? The surrounding of the cancer cell is acidic in nature, and the tumor microenvironment is acidic, and it facilitates the metastasis that is well established. And this concept is utilizing in the imaging. And now this is the simple glycolytic pathway where the each and everyone, you, me, everyone is, our body is following the same way. And here this is, and the beauty of our nature is that they are very, very strict their rules. So if you see here the biology here, this is the step by step sequence. The glucose is metabolized in the cells and they are converting into the energy. So here we can see at the end, LDH is exist at the end. That Chandra Prakash talk about, and the William Laurie talk about the LDH. LDH play a critical role in tumor glycolysis and cancer biology. So here, I want to say here the hexokinase enzyme, that is the first enzyme. That is the beauty of this enzyme. If glucose is transported through the GLUT transporter, here everything is important, transporter as well as the enzyme. So if glucose is transported into the cell, so if it is not phosphorylated through hexokinase, then it is not absorbed. So everything is important and it is strictly followed this one. So as from morning to till time, we are discussing about the overexpression of the enzyme. Here, in comparison to normal cells and cancer cells, these enzymes are overexpressed. Every transporter and enzyme in the glycolysis is overexpressed. Now it's the choice, or we need to study which enzyme is the important to target and which is easy to target. After targeting or designing a molecule, how it work in the system, in cell, in vivo or inside the body. So here I am interested in LDHA. I worked in LDHA during my postdoc and after postdoc. So I am a little bit interested in LDH. Why I am interested in LDH? There is some clue here. So glucose is converted into pyruvate. Each glucose molecules change into 2-pyruvate. And the pyruvate is a hub of energy. Here at this point, in the normal cells, pyruvate transfer to the mitochondria for production of energy. But in case of a cancer cell, it transfer towards the lactate production catalyzed by LDH enzyme. So there is a bifurcation point. We can selectively inhibit LDHA and we can selectively kill the cancer cell by inhibiting LDH. Now this is not a simple task. We can just design a molecule and hit this LDH target and it selectively selectively inhibited and we kill the cancer cell. These are the challenges, and what are these challenges I will discuss in couple of slides. So first to discuss about the challenges, how we are facing the challenges to design a molecule against a target. We must know a little bit about the structure. So LDH is a tetrameric structure where these are the composed of 4 subunit. LDH A subunit and LDH B subunit. So LDH A and LDH B subunit both are encoded by different gene LDH A and B, and by the possible combination of these two subunits, there are 5 isozymes. The LDHA, that is composed of simple LDHA unit, that is homozymes, and LDH4, LDH3, LDH2, and LDHB. So here we can see LDHA and LDHB both are homozymes, means they are composed of same type of unit, and the rest of the 3 are the heterogen. So now, they are structurally similar, but the overexpression of LDHA and LDHB is different. LDHB is overexpressed in predominantly found in heart muscles and spleen, kidney, whereas the LDHA and 5 predominantly found in skeletal muscle. So we can selectively target which type of tumor we want to target here. Another, this because of the structural similarity, to selective target of these enzymes is difficult. Now, structural analysis. Here we need the computational chemist, AI and ML. So we can see here, this is the NADH molecule in green and blue color, and here this is a small, that is the substrate, and this the long molecule, you can see this is the NADH. So in the catalytical mechanism, any cofactor as well as the substrate both play important role. So we must know, before designing a molecule, we must know the catalytical sites, which type of functionality or amino acids are present in our catalytical sites. Based on that, we design the counterpart and we'll check it. So here we can see this catalytical site is full of rich of arginine, that is cationic in nature. So this site is cationic in nature. Another important challenge is this substrate is very small. So we need a small molecule, and this is the cationic. So the substrate site is this catalytical site is polar in nature. So we need polar molecule, we need a small molecule, and the size of this pocket is very small. So to enter a molecule, big molecule into the site is difficult, so we need a small molecule. Here, very small molecule. This is the basic challenge to target LDH. So here, this is the challenge to target the LDH, but there are several molecules identified, natural product as well as the synthetic molecule. The first molecule that is isolated from cottonseed, that is the gossypol. So this molecule shows the LDH5 inhibitory activity is 1.9 micromolar, and LDH1 is 1.4. There is no selectivity, but that is active. But now this molecule entered into the clinical trial, but after third phase it was removed. Why? Because of their toxicity, and their toxicity was due to, I will tell you here, due to this aldehydic functional group. Why this aldehydic group is creating the toxicity? Because this is quite active. When it enters to the biological system, it binds to the enzymes and it creates the toxicity. So this molecule failed. After that, there are several analogs was made by the gossypol, and all the analogs shows very nice activity. Some shows the in nanomolar, but all the molecules fails, in some fails in the preclinical trial and some fails in the clinical trial due to their toxicity. And now I want to tell you one interesting theory here. So the small functional group and a small change in a molecule can create a big change. How? Here you can see the gossypol. I have written here R. Okay, so here you can see this solid bond, the black solid bond here. It means that is up. So if we change these bonds down, means it becomes R. That is totally inactive. This is the beauty of the small changes. Now I will discuss some molecules, those are identified by the industry, and they showed activity in nanomolar range. These are the molecules that is discovered by GSK, a second one discovered by GNE 140 by Gentech, and the compound 22 is again GSK. So these are the molecules, those shows potency in nanomolar activity, but all fails after preclinical or clinical trial. Again, reason, for example, this GSK1 molecule, it has biochemical potency in nanomolar range, but its cellular potency is not good. So it fails. Again, the compound 22, same problem. The GNE 124, it has the biochemical potency as well in cell potency, but in vivo optical, they don't have the PK profile. So we, as a medicinal chemist, we want, as a drug, we want everything. So here in the last compound, you see number 1, it has the biochemical potency as well as the cellular potency, but they don't have the PK profile and in vivo LDHA. Then now the medicinal chemist and computational chemist work start here. So the compound 1, we need to change the compound 1 based on the structural analysis, how they are binding with the enzymes. So this is the change to hit to lead generation, and by changing small things, fluorination and this molecule got the biochemical potency, cellular potency, as well as the optimal PK and the in vivo LDH inhibition activities. Now both molecules are in clinical trial. Now, I want to tell you that the small change, how we can do a small change in a molecule, and that don't think about that this is random. This is not random. This is based on the chemical properties of the functional group. This is based on the computational chemistry, how they are bonding. So here, this I will discuss about the halogens, fluorine and chlorine, very small group, and when we insert that, what happened? So here, these are the drugs that are in the market for different disease having the halogen fluorine. So they are, and this is not making the molecule hydrophilic, they are making the molecule lipophilic, but they are improving the PK profile. So here you can see this 7-chlorocululine, the anti-malarial drug. If this R is H, the activity is IC50 value in nanomolar range. If R is changed into Cl, it drastically change like 37. So its activity increases 29-fold. Again here, you can see by changing the small groups R to Cl, its activity changes 15 times. So that's the things we are changing the activity. I don't want to take because we are running short of time. So here just I want to say after small changing by R to different small fluorine and chlorine group, this activity or potency increases 200 to 1000 times, 3000 times, 1500 folds immediately increase by inserting a small group. So here these are again. Now this is about the potency. Now we can change the pharmacokinetic profile by changing a small group. Here we can see here the IC50 value is not changing after changing this R group. Okay, it's Cl. So we can see here the half-life. It's drastically changed. It's the clearance. It's drastically changed. Then again here, activity is nanomolar range. When we are inserting a group, a small group, I am talking about this, then here we can see the area under the curve clearance is very improved. Here again here, the kinase activity is same, but the clearances and the pharmacokinetic profile is drastically improved. So, and here again the position also matter. So I will just give a clue here. When we are changing this X by group by fluorine, by substituting in different position, what happened? The activity change, but this at the para position here, when we are inserting fluorine, it gives the best result. Why? Because here it creating a bond with the amino acids. So if this is not creating a bond, then there is no change, there is no need to change. So we need to change about thinking about this. The changing functional group can participate in the binding cavity. So again here, after changing various group in different position, the activity improved. Again, the reason is hydrogen bonding and electrostatic interaction. Then another important point, suppose we have a molecule with the nanomolar potency, then we tested it in the cell, we tested in vivo perfect, then we test in the that molecule has the metabolic stability. So what happened, this molecule, that is the agitamib drug, that is the cholesterol inhibitor, but when that drug inside the body, that create the problem, it has the due to the oxidation process, this molecule has limited applications. So after changing these, where I put the arrow, after changing these functionality, this hit to lead change the, you can see here efficacy is 2.2 milligram per kg per d. Now it becomes 0.04 and the is oxidation blocked and its the oxidative metabolic stability increased. Further, again, this is the prostacyclin, and that drug is used for the platelet aggregation. And this is due to this, the failure of this drug is due to this ring, small ring. And what happened? This half-life clearance is very short. So after changing here, applying something here, small changes, this can be improved. So what scientists did, they just CH2, this CH2 is modified with first 1 fluorine, then 2 fluorine. And you can see the half-life is drastically improved. And now this drug is in the market. Then the whole story is behind the small group, and this is the halogen bond interaction, the interaction between the molecule and the enzymes. So I'm not going to discuss because of short of time, and if anyone want to discuss, so we can discuss later. And how the bond is forming. Please, this is the nucleophilic, acidic, and acid and base interaction. That's all. That's all. So now there are some pros and cons of small group interaction, small group installation here. So if we are adding a small functional group in a molecule, their binding affinities not change. Why? Because we are not changing the large group. We just change their pharmacokinetic profile. If we are adding the large group, then it may change the binding affinity as well as the pharmacokinetic profile. So I want to thank you, Professor Filippo Menitolo, where I started work with the cancer cell metabolism, Department of Pharmacy, University of Pisa. Vinit Kumar, sir, he invited me to deliver a talk, and my PhD supervisor from IIT Roorkee, Professor S.M. Sondi, Sushil Kumar, IIT Bombay. Whenever I need some suggestion, I call him, and Dr. Chandra Prakash, AIMS Delhi, Dr. Anil Sharma, IIT Roorkee, Luca Gatinoni, Germany. He provides some input during this program. Dr. Mayank and Dr. Saran to move forward in this area. Thank you all.
Thank you very much. It's a very excellent talk. I will request only students one question because we don't have any time. From students side, any question? Okay, then we will move to the next session.
I request Professor Simran Tandon ma'am to facilitate Reshma. And also Dr. Shinjini. So I would like to invite Dr. Virender Kumar and Dr. Subrata Pore to moderate this session.
Okay. So good afternoon. It's my pleasure to invite Professor Saran Kumar from IIT Delhi. And before he starts his talk, so let me give some brief introduction about him. So Dr. Saran has did his BSc and MSc from St. Illinois College, Mangalore. And then he did his PhD from DBS Singapore, and that's also my PhD department, and we were at the same time almost, and so we know him since PhD time. And then after his PhD, he and moved for his postdoc to the Hebrew University of Jerusalem in Israel, and then he moved back to IIT Delhi in 2021 and joined Kusma School of Biological Science as assistant professor. He is working now, so he has left Israel at the right time, right? So his research interests are like it's quite diverse. Vascular biology, cancer metabolism, tumor heterogeneity, like that. And he has very good publications like in Science, Nature Communications, and nucleic acid research, like that. So so with this short introductions, I'll invite Dr. Saran for his talk. Yes, please.
Yeah. नहीं नहीं ठीक है मैं कर दूँ लेजर पॉइंट नहीं चाहिए नहीं चाहिए क्योंकि वीडियोस में जब प्ले नहीं होते करना
So hi, I'm Saran. So I know it is post lunchtime, and I can also see that some of them have already slipped through nicely, smartly. So it's a polite request. Firstly, thank you to the students who are still there. And maybe I'll ask your Dr. Vinith or something to give one more additional mark if you're still there till I finish my talk. And one more additional mark, if you guys keep your mobile aside for 20 minutes, I will try to cater the talk as much as I can so that you are all interested about the work which I'm going to present. So please, again, and some of you, if you can come ahead and sit, it will be great. So I promise you not to bite. So okay. Anybody, please. Some of you move front and come and sit in front. It's okay. Promise you, I won't take more than 20 minutes. So 20 minutes, you can say 7. Your time is up, and I'm out. Okay. So let's begin. Please, come on. It's okay. Come. I promise you not to bite or not to hit or anything. Come. Not to scold, not to ask questions. Nothing. Okay. Yeah. So it's always nice to have a younger crowd so that whom we can give a talk. So that's the idea. Okay, so as you guys are settling in, I'll move forward because already time is short and I don't want to take too much of time. So again, I'll be talking about metabolic heterogeneity in cancer. So it's a too complicated a word, a complex a word. Don't be intimidated. Let's go ahead and see what exactly it is. And as I speak, yeah. So tumor heterogeneity, right? So basically, we are all colorful. So exactly, the tumor also is like that. So it's not uniform. Everything has different kinds of structures as well as characters. So everything is different. So if you are logically trying to target it with one drug, how will it work, right? So that is the problem the field of cancer has been facing for 50 years. And I promise you, not being pessimistic, but a realist, that it will be the same even for further 50 years. Okay, so what do we do about it? We try to address this in a multi-pronged approach or in a strategy which we can employ so that we can understand this better, so that we can treat this better. So you can see this colorful like a holy of tumor, right? So basically what is it is basically each circles are different tumor sections but are all of same colon cancer tumors. But if you see the color, especially the red and pink, pink and green, you see that some of them have different shades of these colors, right? So again, it states basically that even though they are all same tumors, but from different patients, they have different flavors or different characteristic features. Okay. So now, again, I am sure you heard about a lot of stem cells, cancer stem cells, and everything. It is the same with this also. So there is again a different kinds of a phenotype when it comes to cancer stem cells. Now, why is it? What is the problem with this? Again, as I told you, if this is the case, then it is really difficult to target it with one particular drug, right? So what is my interest and why am I giving you a stage, setting you a stage for this heterogeneity? That's what I will call from now. So I come from a field of vascular biology, which means studying blood vessels for different fields. So I am interested in cancer biology. I am interested in how blood vessels are important so that your hair doesn't turn black to gray. Otherwise you would definitely confuse me with Professor Das, don't you? Almost the same feature and the beard is the same, right? Only thing is he has much more gray cells and also much more intellectual capacity. So again, so blood vessels have a lot of features. So one of this is basically to understand why having these, which are the ones which basically feed, all the cells, if they are different and how are they different and what would they create if they are different. So this is what is I'm interested in. And that's what I'm going to talk about. And so just a very basic introduction. It is one of the major extrinsic factor. Basically, you might have studied a lot of genetics already, right? So any kind of heterogeneity can be because of inherent nature, that is the genetics, or there is a DNA, or it could be just because where it is, like for instance, now we are going into the months of pollutions, right? So we will have a different feature than once somebody who is sitting in Iser Pune or Iser Trivandrum where the weather is so beautiful, right? So these are the things which will affect. So these are environmental factors which are going to affect, So those are the things. And vasculature falls in the environment factor because this is something which is feeding the cells from the outside. If you think that there is one particular cell. And my interest or my talk for the next 15 slides, that's what I will have, 15 slides, is basically on glioblastoma, that is brain cancer, the cancer which affects the brain. And why? Because it's one of the most severe cancers. Once it is detected, usually the median survival is less than a year or a year and a half. Okay, and also there is heterogeneity seen in that. So this particular slide, if you see, there is this tissue, right? So this is a tissue section. You can see nice pink regions. So these are the necrotic codes where there are no cells, and there are these hollow white marks. These are the blood vessels. So what happens is basically blood vessels cannot reach all the places. So it would be like, for instance, okay, for all of you now, I am saying that all of you are hungry. I am going to keep only 10 plates outside and eat. So some of you will be well fed, whoever is strong, whoever is capable, whoever is lucky to get adjacent to the plate. The ones who are farther will have different kind of features, right? They'll be super mad, super angry, right? So that is what is happening. And how do we study this in cancer? So that is what I'll try to show you in what we have done. So the question, a scientific question is, how does perfusion? So basically, when the blood vessels are basically the conduits or the pipes which give a lot of factors as well as oxygen, nutrients, etc. So how do they influence tumor heterogeneity? So the problem in the field was that there was no proper appropriate tool to observe or understand this. How do you basically locate somebody? Like, we wanted some kind of a GPS map or a location. How you say basically in WhatsApp and I have to reach you, basically you just share your location, right? So we wanted to do exactly the same. So what we did is basically we took the cancer cells, which is GFP tagged. So basically these are color coded. Why? Because cancer is not just cancer cells. Tumor is not just cancer cells. It also has a lot of immune cells, other infiltrates, right? So we wanted to study first only the cancer cells because that's what we are interested in targeting at the beginning. Then what do? Basically, we use a mouse model. So the cute mice which you see is basically immunocompromised mice. That means you can inject human cells into them, okay? Because we want to study the humans, it can affect mice, but who cares, right? Then what do? We implant the tumors into the mice, do something called as a vascular perfusion. Once the tumor grows in the brain, you see, this is the tumor which has grown in the brain. Then what do? Because the circulation which is there, it connects from heart to all over the body, right? So it will reach the brain also. So what we do is basically we take a dye and perfuse the dye through the system. So basically we flush it in through the system with the logic that it will go to the every cells or almost all the cells. This happens in your case every day. Every cell of yours almost has the perfusion limit, so it is happily surviving. The time if it doesn't, then there is problems. Like for instance, in amputation in the case of diabetes, why is it? Because ganglion, what is it? The blood vessels are not able to reach there, and then the cells are dying, and then you have to amputate. So that is why it is. Okay, so same principle. So we give a dye, but only the difference is that this is a cancerous model. Once you give the dye, it is going through the blood vessels, okay, and you resect the tumor, and then you dissociate them into simple single cells. So basically chop it off. And then what happens? You use a salter. I don't know if you know what is salter. So basically it will help cells to sort based on the dye uptake. So what is the logic? If you were, let's say, again giving the analogy of the food and the plate, if we're closer to the plate, I could have snapshot you, took a picture, so I know that you are the one. So basically the salter will say, okay, those with the plate and those without the plate, please move aside, and you do. So that is what is a salter. And you do that, and the cells which are picking the dye will be the ones which are closer to the blood vessels. The ones which are very, very, very far, they cannot uptake the dye because we give exactly that much amount of dye so that they don't reach there. Okay. So then what happens if you see this particular slide? This is a tumor slide. I don't know. So see this one. This is basically the green color is all tumor, right? We injected a green cells, you remember? And then this red one are the blood vessels. So basically blood vessels, if there's a pipe, if you chop it off, you see the cross section, you see a round color thing, right? So that is what it is. And then it distributes. And then what we can do is, based on the dye intensity, you can really say, okay, give me which has taken 100% of the dye, or 80% of the dye, or 70% of the dye. The logic being, how far are you from the blood vessel or the functional blood vessel? Make sense? Make sense? Okay. So then we did that. We took 3 fractions. Okay. From now onwards, I'll call it as high, medium, low. The high means it is closest to the blood vessels. Medium means little far. Lower means farthest. Okay. HML. So what did we do with that? The idea was to isolate these cells, and then we could do multi-omics. So basically that means we could get the whole history of the cells in terms of what are the RNA which are expressed, what is the kind of protein that are there, what is the kind of metabolites that are there, all could be done with this particular approach. So that is a technique which wasn't there, and I did it. And if you allow me, I can show you a cool video if it works. I don't know. No, sorry, it doesn't. For some reason. Is somebody from the can help? No? It's okay, leave it. So basically this was a 3-dimensional image. It showed the how this tissue section is there, and then there are this blood vessels, and there is this perfusion dye, which is blue color dye. That's okay. So what did we do with that? Again, I told you, right? High, medium, low. So we had 3 different this one, and remember, these were all identical cells. Okay, beginning it was a clonal population, and just that where they were present was different. And because of that, if you look at this heat map, you can clearly see there are 3 different signatures, right? This belongs to one group, this belongs to other group, this belongs to other group, right? So these are the 3 different subpopulations just because where they are present, and the gene signature is very different. Once the gene signature is very different, that means they are very different, right? So that is the logic. And we could do both transcriptomic, that is RNA studies, and metabolomics, that is the small metabolite studies. Then what we went on to do is basically, now we want to understand how basically the blood vessels are shaping these cells. What are the kind of influences that they have. So we did certain pathway analysis and try to understand what are the ones which are expressed, and etc. etc. So in short, it is basically, again, going back to the same analogy of the plate and the food and the students, the ones who got the food were the happiest, right? And the ones who did not were the ones who were sad, right? So similarly, the perivascular cells, or the cells which are closer to the blood vessels, had a anabolic signature. That means, like, I can sit through more talks, let's say, or I can do some more activities. That is the kind of a setting. So we did a lot of studies and tried to prove that basically that these cells which are closer to them are indeed having a different anabolic signature. That means they can synthesize more proteins, okay, and they can do more of a mitochondrial activity, okay. And remember, right? These were all supposed to be the same cells, in vitro when you grow it, it is all same, just that because they are different, they are behaving very differently. So we did series of tests, and we showed that thanks to a lot of people who have already talked about Warburg effect and how glycolysis is important. But here we saw that the cells which can go for, let's say, mitochondrial oxidation, thanks to Dr. Chandra also, which he supported in a way, and can go undergo this one. So basically, the cancer cells are smartest. Based on kind of resources that you give, they will take it. So basically, again, going back to the analogy of your food. So there are 10 plates. Those who did not get, maybe will go and search for samosas which were still there from the one which was left during the mid chai break, right? So that is the same logic. Okay. Now, let me just rush through. So what else did we do? So we went in to check whether what is this anabolic signature, why is it happening? So one of the major mechanisms how the cell senses if it is hungry or not is basically through mTOR signaling. Okay, if you give them food, then it knows, it will tell the cells that, hey, listen, there is food, you have to produce a lot of things, keep building things for yourself, because you don't know when the person will again go and sit in another seminar, and I don't know when you will get the food. So when you get the food, try to metabolize as quickly as possible, right? So that is what has happened. And we saw that mTOR is indeed active. There are certain amino acids which, when the cell senses, it says that, okay, I have a lot of resources, turn it into something worthwhile. Okay. And then also we checked with this with respect to how it is happening. So we had some certain hits in the transcriptome. So we did a complete analysis of all the genes, right? So we saw some are up, some are down. So we validated these genes, and we found that some of them are indeed inhibiting, like REGT1 to TSC1. These are all, let's say, just inhibitors of mTOR. They were far away from the blood vessels. The red is the blood vessels, right? So, and the white patches are far away. Whereas phospho-6 and phospho-AKT are actually the activators of mTOR, and you see that they are the ones which are active near the blood vessel. Same, these are all till now I was talking about was still in mouse model, right? So now we took it one step further, and we saw that even in human samples, it was the same. Okay, so these are all human patient GDM samples, and we resected them and we checked for the staining, and it was the same. So we indeed saw that near the blood vessels, this do happen. So now I have convinced you that we have a new tool through which we can really study what vasculature does in cancer. And this tool was not just to study, but also we could take these cells, retrieve these cells, and do some functional assays. So we did series of functional assays to show that the cells which are closer to the vessels are indeed aggressive, are indeed having a lot of functions. So for the because of the time, I'll just skip it, and we performed even lipidomics, that is to look at the membrane, how they are different between these cells. Okay. And what is the takeaway message? Okay, they are different. So what's the big deal? The big deal is that they have a different feature or different sensitivity towards a drug. Okay, so that is what we found out, that if you give a particular drug, let's say a TMZ or a irradiation which is given for the GBM patients, you see that the cells which are really enjoying the blood vasculature have a kind of a resistance, and that can be really nullified if you stop that particular mTOR pathway which I was talking about all the way. Right? So this is the translation message which has come out of this particular study. Now, again, if you just go back one step back, these are all same clonal population, so they all have the same genetic map, right? More or less. Now, why is it that they are behaving very different? Yes, they have been fed differently, but what is the rewiring that is happening within? Because they have all the same genes, right? So if they have all the same genes, why is it different? Then it is because of the epigenetics which happens. Either it could be chromatin modeling or it could be methylation. Okay, just read a bit about these things. So these are the ones which makes even a person, once he's there, let's say, like, I was talking about how a person ages. So these are all because of the different methylation, different epigenetic changes which happens throughout the age as accumulation. It is not necessarily always mutation. So because we have this cool technique, we thought we could also do this. So we did an epigenetic analysis, and we found that again, this hole is basically a blood vessels. These are blood vessels, and if you see this cute magenta color, which is there, which is far from the cells which are closer to the blood vessels. Okay, so they have a different methylation pattern. So that's what we found out, and with Professor Rahul, who is sitting there, we met somewhere during a sports meet, and we had a good collaboration, and this result is out of it, wherein we thought, okay, why he is a computational person and I'm a bio person, so we thought, okay, why not take this forward? And I gave him the data, and he analyzed, and he saw that indeed these cells have a different epigenetic features. Okay, that is what is keeping it as a memory and saying that, okay, you behave differently, you got food, you did not get food. Okay, and we saw that, and we have these cool heat maps which show the same, because we did an array for 850,000 CPG islands. So basically, wherever there is methylation in your genome, we scanned all of it, and we could come up with this. So this is one of the first spatial methylome which has happened, because of the influence of perfusion. And this is yet unpublished, so I'm just presenting it to you. This is the first time I'm presenting it, so you are this one for me, inspiration for me. So thank you for coming in the front. So then what is the differential perfusion leads to? So what does it do? It changes the methylome profile. Why? Because what happens, every gene to get activated, there is some transcription factor, something coming and binding, right? So these methylations disturb that or promote that. Either of the two can happen. Okay. So specifically in the promoter regions of the gene, let's say for instance here, if you see, if there is already no methylation or methylation, if there is methylation, then there is no gene expression. If there is no methylation, then there is gene expression. Okay, so that is the idea. So you see that there is a clear-cut difference in the methylation regions, and specifically in the promoter regions, there is an enhanced hypermethylation or hypermethylations, especially in the promoter region. That means the genes which are important are being changed to do the tasks based on the kind of resources they have, right? That is the vasculature. So we did some more detailed study to combine the transcriptome and the methylome and say, hey, listen, which are the ones which are really making sense? Are there something which can come out of these? So these I will be happy to discuss maybe in the next coming conference which Dr. Vineet will organize. So till then, stay tuned. Thank you so much. One second. Yeah. Sorry, this is my acknowledgement slide. I have to put.
That was an excellent talk, sir.
Thank you.
My question was, so we all know that there is the hallmark mark of any cancer is neovascularization. It produces new blood vessels. So have you considered the possibility that the cells that were far from your blood vessels, can't they neovascularize?
Very, very good question. So, which for the sake of time, I skipped it, but yes, good first point. The hallmark of cancer vasculature is one, yes. And second, what we did is basically not just about vasculature, we went on to search for perfusion. So, when I do this, like let's say when I perfuse the dye, it will take care of all the new vessels or also which are forming, which are functional, because I'm perfusing the dye. So, if the tumor has already started forming new vessels, it would have already created new vessel, the dye will go there and it will mark that it is perfused region, so no worries. So, but this is a very good point. Yes, we have considered this, and what I'm still interested in, still there are regions which are completely necrotic or completely hypoxic, and then the whole concept of this vascularization triggers. This is a very good question. My supervisor would be very happy because he was the one who found out that blood vessels start coming in GBM during that time. Thank you. Good question.
So excellent talk, very eye opener. So
thank you.
So this very basic question. So you found some chromatin methyl transferases are altering in the high and low population.
I am not an epigeneticist. You are the epigeneticist. I am coming to you for asking regarding that. On a serious note, we have just looked at these genes with respect to that. We are looking at methyl transferases, DNMTs, and others. We haven't done a comprehensive study because first we would like to put this out with respect to validation of some of the genes. But Oh, definitely would be happy to discuss. Yeah, over lunch. Otherwise, yeah.
Okay, so Dr. Sen will be here during lunch, so the students can ask questions that time also. So please join me to thank Dr. Saran for his excellent talk.
Yeah, thank you all.
And I'd like to request Dr. Prakash Waligar and Minu Kumar sir to come up to felicitate Dr. Saran. Good afternoon. Let's move on to our next speaker, rather last speaker, Dr. Mayank Singh. It's my pleasure to introduce Dr. Mayank Singh. He is an associate professor in the Department of Medical Oncology at AIMS, New Delhi. He has more than 20 years of research experience in the field of cancer biology from different institutes like Ohio State University, Cleveland Clinic, and the University of Texas Southwestern Medical Center. Dr. Singh published more than 50 research articles and received several awards. His active research areas are ubiquitin biology, liquid biopsy as a diagnostic tool, development of CAR T cell therapy for the treatment of relapsed and refractory multiple myeloma patients. And now I'd like to request you to deliver your talk. Thank you.
Pointer share. पॉइंटर है अच्छा ठीक ठीक thank you organizers for inviting me here and I'm the last person before the lunch, so I will try to make this talk as much interesting as possible. It's actually a different talk as well what you have been hearing since the morning, and you are hearing a lot about the lot of metabolic pathways and all those things. So I will like to switch gear, and I think I will like you to discuss something about the immunotherapy, because we have been talking about the cancer since the morning, and we know that cancer is like it's a very heterogeneous disease, like Saran was talking to about right now. It's a very heterogeneous disease. The cancer cells, they keep on erupting, and you throw anything at a cancer cell, you throw anything, it's eventually what is going to happen is that it's going to develop resistance against it. So across my 20 years, I've been working in cancer research more than 20 years now, and what I realize is that you need something what we call as a continuous immunosurveillance. You need some form of a live drug which can actually keep a tab when the cancer cells are growing, and actually they can kill the cancer cells as they grow. So keeping all this in mind, I decided to present this work, which is on the CAR T cell therapies. And CAR T cell therapies, I don't know how many of you have heard about that. So these are basically chimeric antigen receptor T cell therapies. So these are basically live cell therapies which we talk. And CAR T adoptive cellular therapies reimaging immunotherapy to combat cancer. And what has happened across, I think, since 2017 is that these therapies really come up a long way, and they have give really good results in the cancer which are refractory, which was not responding to the drugs, and they have come up. So I'd like to start briefly by giving an introduction. So what is actually the immune system? As we all know, the immune system recognizes and reacts against cancer by regulating the immune response. And the cell-mediated, which we call as cell-mediated immunity, is the key player that generates response against the tumors. But what happens is that the cancer cells, they evade these immune response, and it is one of the hallmark of most of the cancers. So the CAR T cell therapy, what it does is that basically it train your T cells to recognize those cancer cells and search for them and kill those cancer cells. If you see the evidence of immunity against tumors, obviously there was enough evidence to represent that. Obviously, that some form of immunity is present against a tumor, but the immune response which is present is pretty weak. It's not working that well, and also all component of the immune system, non-specific, humoral, and cellular, can affect the growth and progression of the tumor. But what happens is that in the case of cancer is that when a cancer cell grows, there is an escape from immunosurveillance. And how does this happen? This happens that tumors may not express neoantigens, means the antigens which are required to target, and tumor may fail to express the co-stimulatory domain, because if they don't express those domains, you don't get what we call as an optimum activation of the immune system. If you don't get the optimum activation of the immune system, then the cells survive. And obviously, certain tumors are known to lack or express these poor neoantigens. So, keeping all these things in mind, I think I will straight away I'll skip to the what actually is the CAR T cell therapy. And if you see the CAR T cell therapy, CARs are basically genetically engineered fusion proteins. So what exactly these are? These have two components. It is a chimera. So chimera is composed of two different parts. The first is your antigen recognition domain, which is derived from a monoclonal antibody. It can be any monoclonal antibody against any form of antigen. Generally, the most famous ones are the BCMA, the B cell maturation antigen, or the CD19 CAR. And then you have intracellular T cell domain, which is called these ones. Okay, these ones, and these are the external domains which basically recognize your antigen, and the lower the intracellular T cell signaling domain, which basically conducts the signaling which kill the cancer cells. And if you see the mechanism of the CAR T cell therapies, you have these domains which are recognizing these. You have the co-stimulatory domains, which is CD3 zeta domains, and once these CAR T cells, which are armed with a CAR, they go and recognize the tumor cells, they go and bind it, and then induce the what we call the targeted cells apoptosis. And CARs have got respective advantages and disadvantages. Like they have a flexibility and dynamic range of CAR is attractive. The current CARs are limited to recognizing cell surface antigens. But the most important thing is that the CAR do not require antigen processing. So you do not require any HLA typing at all for this one. So that is why it can be rapidly developed also. In fact, there is a crux on the development of the shelf CARs also. And CAR are more secure, better targeted, and much more persistent. If you see the different generation of the CARs, like first generation of the CAR, the first generation of the CAR were designed like somewhere in 1980s, but they were not that good enough. They were not that good enough because they do not have a proper co-stimulation. They were able to bind, but they were unable to induce what we call as the apoptotic signaling. So then came the second generation of CAR, which has this co-stimulated domain, which we call as a 41BB or CD27 domain. So what it was doing is that we were getting an optimum activation of a T cells, which was causing the killing of the cancer cell. And the key advantage confirmed by the use of second generation CAR was the introduction of the secretion ligands, which were able to stabilize the tonic signaling in these ones. Then we have the third generation of the CARs as well. So in the third generation CAR, what happens is that you got the additional domains, which are additional co-stimulatory domain. But what happens is that this is calling an aberrant activation of the immune system, which is related to severe what we call as a cytokine release syndrome. And because of the cytokine release syndrome, you're getting a lot of toxicity, which are uncontrollable in most of the patients. So that is why the most of the CARs which you have in the market today are the second generation CARs. And this is the pictorial representation of one of the articles she published, in which I'm showing the first generation CAR, the second generation CAR, which has got multiple domains, the third generation CAR, which has got 3 different domains. Then we have got the other generation CAR, which has the fourth generation CAR and the fifth generation CAR, which are basically currently used in the case of the solid tumors. But you have the cards, but you also need a good vectors to develop the card. Once you have the card in your hand, you need something to transfer these one into the T cells. So generally, the most popular the vector which we use, which we are also using in our lab, are the lentiviral particles, which are the basically viral particles. You synthesize your CAR, you encapsulate between your lentiviral CAR, and then you transfer those that. But there are another popular way, like ZFN, TELN, CRISPR-Cas9 are also being used. So this is a typical flow chart that how it is done in the patients. So you have got you can extract from the blood, you extract the T cells. These T cells, then these are these what you see here on the surface of these T cells are the CARs, which is called. Then at this point, what we call them as the CAR T cells. Then these CAR T cells are put in back to the patient, where they go into the patient and then they target the tumor, and the tumor is killed. That is a particular flow chart of the CAR T cell therapy. So why CARs? Because best of both the world. They have the B cell specificity, T cell cytotoxicity without presentation. It is a form of adoptive T cell therapy, and it targets surface molecule into the native conformation and in engaging target independent of MHC complex. That is the biggest advantage of the CAR. Ideal CAR target will be tumor specific, universally expressed only on tumor cells, cell surface molecules. The most common target as of now is CD19, against which the FDA CAR has been approved. The BCMA, against which we are also developing the CAR. And these are the other targets which are present. You can go to this article and see it in the whether in BELL, TLL, AML, and these are different in clinical trials which are being developed. And if you see the FDA approved CAR T cell therapy timeline, if you see in 1993, the first generation CAR was seen. And after that, you have the second generation CAR. And in 2014, the first human BCMA CAR T cell therapy clinical trial was initiated. And in 2021, FDA approved ID cell for multiple relapsed and refractory multiple myeloma. And if you see the current landscape of the CAR T cell therapies in the market, you've got four products which are approved against CD19, which is against Tamra, which was the first one, obviously. And then you've got the two products which are approved against the BCMA antigen. So what I'm saying is that they are approved against a target. Okay, target is expressed in any cancer, is going to go and kill it. Like in the case of BCMA, this one is approved against relapsed and refractory multiple myeloma. It means the patient, if no therapy is working against it, it is refractory against all the drug, then they are approved for that. And the clinical trial results of these CARs are particularly amazing. But these are the CAR T cell are also associated with complications. The most common complication is your CRS, which we call a cytokine release syndrome. It is typically within 5 days, and it is exponential T cell proliferation, which lead to activation of your what we call as inflammatory markers. And obviously, it is controlled by using tocilizumab or other steroids on the line. But if you see this one, this is a figure which we have taken from the article which we have published recently. So CAR T cells have got different challenges. Like what I've been talking about till now is that these are different CAR T cells which are approved against different so-called blood cancers, like your like ALL or multiple myeloma. But we don't have as such the approved CAR T cell therapy against what we call as solid tumors. The reason we don't have against solid tumor is that because there is heterogeneous expression of the antigen. The antigen is very important. The antigen has to be specifically expressed in your target. If it is expressed anywhere else, it can cause a cytotoxicity. The CRS, I have already discussed it, and obviously your tumor-associated antigen escape, which happens in that. Then in the case of solid tumors, what we call what present is that an immunosuppressive tumor microenvironment. So what happens is that you have a solid tumor, then you have a core of a tumor in which there is a lot of hypoxia. In this hypoxic core, the T cells are unable to work. So that is why they have tried to develop CARs which have like HIF1 alpha-inducible CAR, hypoxia-inducible factor-inducible CAR. So these CARs can go there and get activated. So these are different strategies which are being used to develop the CAR T cell in the case of solid tumors. So coming to our work, we have been working in the direction of development of the CAR T cell in multiple myeloma, which is an aggressive bone cancer. It originates in the bone, and from it goes to develop. If you see the phases of multiple myeloma, it goes from bone marrow, which is called MGUS, is called mononuclear glomerulopathy of unknown significance, goes to smoldering multiple myeloma, then multiple myeloma, and finally plasma cell leukemia. And what happens is that when it does that, what happens is that this antigen, which we call a BCMA, B cell maturation antigen, the expression of this antigen goes increasing in multiple myeloma. So that is why it is a very safe target. It is not being expressed on the normal tumor, but on the on the bone marrow on a multiple myelomas which are being produced. And BCMA, the ligand for binding is BAF and UPREL, and it goes and bind it and activate the signaling pathway. So that is why this is one of the targets which we are working to develop the CAR T cell again. And if you see the landscape of immunotherapies against BCMA, there are many clinical trials which are going, monoclonal antibodies, antibody drug conjugates, the CAR T cell therapies which are being developed against multiple myeloma. And this is a particular landscape of different, this is like old slide, I think 3 years back. If you see that all of these, they're like NCI, UPenn, Bluebird, which is the card they were developing, and all of these, the co-stimulator domains was 41BB, 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 41BB. So this is like a safe co-stimulator domain, and which is one of the co-stimulator domain which we have integrated in our card design as well. And these are the results of the Karma trial, which was done, and it was seen there was a 72% overall response. 72% overall response is a very good overall response in the case of patient who does not have any any drug working over it. And that is why we decided to develop this. This was the DBT project which we are doing right now, and we are working for the development of the CAR T cell therapy. And these are the particular objectives. So what we are doing is that we are doing the generation of BCMA specific CAR. We try different ways to do that. We try to go for the public domain, but we were unable to find anything in that. Then finally, we stick to the mouse hybridoma technology for the generation of SCFEs. Then we have gone and developed these CARs, and we have done the lentiviral encapsulation of these CARs. And now we are about on the objective 3, in which we are trying to do basically animal studies to see the tumor regression studies in these ones. And this was the thing which we do. What we did in that, this is the approach we did. We use the mice hybridoma technology to generate our antibodies. And this is the sequence of the CAR which we are in. If you see, this is the CAR which we are developing. It has got a 41BB CD3 zeta domain, which is a particular of a second generation CAR. And then you got a hinge domain, and this is your SCFA. This is your SCFA. This is an all important domain which we have developed the antibody against, which we have on the process of patenting also, and and our CAR is against S1. So first step what we did was that we first isolated the BCMA proteins, BCMA sequence from the patient who were coming at IHCH at our center, and we collected the blood from the patient from the healthy control as well as from the patient, and we saw that the BCMA level was high in these patients. So then we went ahead and we sequenced this BCMA, and what we did out of that, we found that serium BCMA expression was high in multiple myeloma patients. And after that, what we did was that we went and cloned the whole BCMA as well as just the ECD domain, which is called extracellular domain, because we were developing against a target which was extracellular target. So we just cloned the whole BCMA as well as the ECD. The data I'm showing is for the extracellular domain. So we isolated it. This is the isolation which what we did, and after that we cloned this vector, and we subsequently did the ECD purification. And if you see, this one is just the Western blot analysis of the protein which we did. It was a 42 kilodalton protein, and this one, so we were able to isolate it. And after that, using the XP290 system, we were able to propagate this one. We were able to get loads and loads of ECD protein because we required this protein for the immunization studies. And after that, this is against the SG speech and the Western blot data for that. This is against the his tag. So we incorporated the his tag into these BCMA ECD domains. And after that, we proceeded ahead for the HPLC-based purification of this protein. So finally, we had a his tag ECD protein, which we purified further. And this protein, this is a sequence coverage analysis of our BCMA ECD protein. And after that, we injected this BCMA ECD protein into the mouse, and we generate hybridomas using this flow, the flow chart which I am showing you. And once we did that, and after that we got 20 different clones. These are the 20 different clones, hybridoma clones, which we got out of it. And we did the ELISA-based studies to understand the binding of this protein. And out of these, what we found was this clone 9C4 got the highest binding ability for the BCMA, which we did through different manners. And these are the culture of hybridomas which you see. These hybridomas are basically antibody producing cells, which are basically producing these antibodies. And we produce these antibodies, we characterize it, we did the BL analysis of these antibodies, of the 9C4 antibody. And again, if you see these antibodies which we have purified, we did the binding affinity against the protein which we have purified, which was the ECD protein. And After that, once we complete all this analysis, we went ahead and we did the molecular docking analysis of the anti-BCMA, our 94 antibody, to characterize the unique SCFA domains. So these are the 4 different domains. And in these 4 different domains, the good thing is that we found that the 3 domains have got the binding domains which are common to the already existing commercial antibodies. But one of the domains was unique to our antibody. So we are currently in the process. We have already filed for the patent for this antibody, which through SCFE, which we have taken out. And this SCFE, which we have taken out of this antibody, so this is our CAR lentivector, and this is our CAR, and we have incorporated this CAR into this lentivector, and we currently have this in the lentiviral backbones, and we have generated the lentiviral particles out of it, and this is our flow cytometry analysis. This is not on the T cells. This is on the HK293 cells, in which we have infected these cells with these lentiviruses, and we have found the expression. This is the non-lentiviral expression vector which we have seen, but by that we are able to see about 10%. Currently, we are doing the lentiviral expression, standardizing that part, and seeing that how it's going. So the stage which we are in as of now. So we have just generated our CAR T cells. So now we are planning to do the xenograft experiments, and once we are able to do our xenograft experiment, after that we can proceed ahead with our work on that. So what are the long-term targets of this particular work which we are doing? At the end of this project, we aim to develop a preclinical second-generation CAR, which we have done that, targeting BCMA antigen multiple myeloma cells. We intend to patent the indigenous CAR at the end of this project, which we are doing right now, and subsequently validate in the separate phase 1 study at AIMS Delhi, which we are going to do that. And developing a pipeline for the future CAR T cell therapies. I'm talking to Dr. Vineet also. We are going to ICMR to most probably get some GMP facility if we can, so that we can put this CAR into that GMP facility and we can do the production of all the CARs there, except this one also. So obviously, what the conclusion future perspectives, CARs have a natural T cell characteristics, very powerful, and for the reason should be directed towards cancer cells. The cost of CAR T cell therapy in US right now is $200,000. So even a substantial reduction might be out of reach for many patient area. But we think that if we are able to develop a GMP facility in AIMS, for specific, we can bring this cost down to 10 lakh, and most probably make it very, very affordable for the patient who come there. And successful clinical exploration of CAR in countries like India will critically depend on bringing the cost down to these therapies, and that is one of the target. And that is why we are developing what we call as an academic CAR. This is my small lab. This is Dr. Bunjani Dagar. She's working on the postdoc. Ashna and Ravi, my PhD student. And I would like to thank Professor Kalpana Luthra, Biochemistry AIMS, for all the help she did. And these are all my collaborators. Thank you so much for the patient learning. Thank you so much.
Any questions from the students? The audience?
Excellent talk. Just had a curious question. Your lentivirus approach and everything, right?
Yeah.
Have you, now that the CRISPR-Cas9 has started this one, have you tried to integrate, or was it before that? And
CRISPR-Cas9 system, we would like to go there.
Yeah.
Obviously, we like to go there, but not as of now.
Just thinking of the approvals, I don't know how it is going to. So
I think lentiviral is particularly safe route in this one, as versus like even if you see the trials, the Karma trials, and all those things. In that, they have used the lentiviral based approach only.
Yeah. Thanks.
Can I ask one question?
Yes, please, sir.
I think, Doctor, wonderful talk, and I think this CAR therapy, cell therapy, has gained traction post the award of Nobel Prize. And to my little understanding, I think in West this is being deployed. Would you mind just sharing where are we in India, and likely timeline when we would be able to deploy in patients in India?
Yeah. Okay. So I guess there's already started with IIT Bombay. I guess there's a group there which is already working on CD19 CAR. So CD19, they have already put it into clinical trial there, but I guess the costing of it is pretty high still now. I think it's somewhere like 45 lakhs, something like that. Except that we have some commercial CAR which is coming to India, but because they are commercial CAR, the costing of them is pretty high, and they're limited to private hospitals. As such, in the public hospital, nothing is happening as of now. I think the world has really moved ahead as far as these immunotherapies are concerned. And we are like very far back. And that is why we think we need to be more because right now what I'm talking about here is a prototype. I'm talking about a single BCMA targeted therapy. What the world is talking right now is the bi-specific CARs. Okay, we have a problem already of CD19 negative relapse, BCMA negative relapse. So those are things. But yeah, I think surely we will be there. I think
no, certainly not. We will be there. We must be there for that matter, because if you really see the government, emphasis on biomanufacturing, ultimately it will dovetail into this also at a later stage.
Right,
so certainly we must be there. Okay, thank you. But a wonderful talk, because really an emerging area. It was wonderful listening to you. Thank you.
Thank you. Thank you. Thank you.
I just had one question. Like, according to theory, they're specific, right? CAR cells. So according to theory, the rate should be 100%, right? But the result was 72% in that.
No, that is we were talking in terms of survival. Okay, so the result is supposed to be 100%. That is why you have a CRS. Okay, so CRS, what happens in CRS is that when you give a CAR T cell to a particular patient, it is going anything you develop against. Say I developed against a BCMA. So any antigen, any cell in a body which is expressing BCMA, it's going to go and kill it straight away. So result is there. It's 100%. It's there. But what we are talking here in that clinical trial was in terms of overall survival. Okay, so what happens sometimes that you treat a patient with a CAR T cell, but he dies because of this high CRS. Okay, you are unable to control the interference and all those things. As a result, it develops a catastrophic reaction, and the patient dies. So that is how it is. So that was overall survival. That is, but generally, CARs are like they are going to kill it because they designed that. Right.
Okay. Any more questions? Okay. Then I request Dr. Joshaman and Dr. Virendra Kumar to felicitate Dr. Singh. Please come up.
Thank you.
Thank you. Okay. So now time for vote of thanks, and I'm requesting Dr. Sanjini Dasgupta to do the job. Thank you.
Okay, so good afternoon, everyone. It's my great honor to deliver the vote of thanks as we conclude today's International Symposium on Cancer Metabolism and Drug Discovery, the role of AI and ML. So on behalf of the organizing committee, I would like to begin by expressing my deepest gratitude to our visionary founder president, Dr. Ashok K. Chauhan sir, for his continued leadership, which has always motivated us. I also extend heartfelt thanks to our Honorable Chancellor, Dr. Atul Chauhan sir, for his support and guidance. I would like to thank our esteemed Vice Chancellor, Professor Balwinder Shukla ma'am, for her insightful address that emphasized the significance for innovation, cancer research, and education. A special note of appreciation goes to Dr. Selvanmurthy sir, President of ASTIF, for his valuable presence and motivating words. Moving on, I am immensely grateful to our distinguished speakers, Professor Lowry, Dr. Rahul Kumar, Dr. Chandra Prakash, Dr. Reshma Rani, Dr. Saran Kumar, and Dr. Mohan Singh, whose groundbreaking research and presentation today have enriched our understanding of cancer metabolism and role of AI in advancing drug discovery. I would also like to thank and acknowledge the continued support of our department leaderships, especially our chairman, Professor Das, our directors, Professor Shubhajit Vishwas and Professor Simran Tandon, whose guidance have been pivotal in seamless execution of today's event. Finally, I extend my thanks to all the participants, attendees for your enthusiasm. Last but not the least, I would like to thank the IT and the media. So thank you once again, and we look forward for more such productive collaborations and discussions in future. Thank you. So I think thank you is the magical word. So thank you, Dr. Sanjini, to spread magic through the vote of thanks. And thank you once again, thank you all of you for making this event successful. Thank you.